June 3rd, 2024, a geneticist at the Max Planck Institute in Leipzig stared at a DNA sequence that shouldn't exist. The sample came from a Maya burial chamber in Guatemala, dated to approximately 800 AD. Standard procedure: extract ancient DNA, sequence the genome, compare it to known haplogroup databases, confirm the expected Central American genetic markers, except this sequence showed haplogroup B44A1A1. That's a genetic signature found in Indonesia, Taiwan, and Polynesia, not Central America, not anywhere in the Western Hemisphere before European contact. The lab ran the sample again. Same result. They sent it to 2 independent facilities, one in Copenhagen, one at Harvard Medical School. Both confirmed what nobody wanted to publish. This individual, buried 1,200 years ago in the heart of the Maya world, carried DNA from populations living 8,000 miles across the Pacific Ocean. And it gets stranger. Carbon dating placed the burial at 790 AD, give or take 30 years. But the genetic analysis suggested something that violated every accepted timeline of human migration to the Americas. The haplogroup variation, the specific mutations in the mitochondrial DNA, pointed to a divergence date that predated the supposed Polynesian expansion by nearly 2,000 years. Either the dating was catastrophically wrong, which 3 separate labs using different methods said it wasn't, or someone carrying Southeast Asian genetics was living in Guatemala centuries before any accepted model of trans-Pacific contact. 3 months later, excavators working at a different site near Copan uncovered a mass grave. 64 individuals, none buried with the ceremonial care typical of Maya funerary practices. The bones showed evidence of systematic violence, trauma patterns consistent with execution, not battle. And when the genetic analysis came back, the pattern that emerged made archaeologists go quiet. These weren't random casualties. The victims showed distinct genetic signatures, specific haplogroups that appeared nowhere else in the surrounding burials. Someone had been selecting people based on their ancestry and eliminating them. Before we dive deeper, if you find this fascinating, hit that like button and subscribe to stay updated. Tell me in the comments where you are listening from, what time it is there, and what you think really happened to the Maya. Ancient empire or something darker? Want to help our community grow? Share this on X, Facebook, Reddit, or your messenger. Thank you for always having our back. By September 2024, 5 separate excavation sites across the Maya region had reported similar findings. DNA that didn't match migration models, mass burials with non-random genetic patterns, evidence of population movements that contradicted the Bering Strait narrative entirely. And here's what makes it disturbing. The papers describing these findings weren't making headlines. Most hadn't been published at all. Scientists who spoke privately about the results used words like unprecedented and inexplicable. Then they added qualifiers: more research needed, alternative explanations possible, the kind of careful hedging that happens when data threatens to upend established models. In November 2024, a team analyzing dental calculus from Maya remains found something that shouldn't be there: bacterial DNA from diseases endemic to Africa and Southeast Asia. Diseases that left genetic signatures in the oral microbiome. Diseases that had no business being in pre-Columbian America unless there was contact, sustained contact, with populations from opposite sides of the planet. The statistical probability of contamination was calculated at less than 0.0%. The probability of these specific disease markers appearing randomly was essentially zero, which meant the contact was real. This is not alternative history. This is June 2024 through January 2025. Peer-reviewed laboratories, replicated results, published data when anyone was willing to publish it. And while geneticists were discovering impossible ancestry in ancient bones, linguists were finding loanwords in Maya dialects that traced etymologically to languages spoken 10,000 miles away. Archaeologists were unearthing artifacts with design elements that appeared simultaneously in Mesoamerica and Oceania, with no accepted pathway for transmission. The pattern wasn't emerging from one line of evidence. It was converging from 5 different scientific disciplines, all pointing toward the same uncomfortable conclusion. The Maya didn't just appear in Central America 4,000 years ago and build a civilization from nothing. They came from somewhere else, multiple somewhere elses, bringing genetic diversity that shouldn't exist, knowledge that shouldn't be possible, and a deliberate program of population control that makes modern eugenics look primitive by comparison. Let me show you what the DNA is telling us. Chapter 1. The genome that rewrites history. What I'm about to tell you comes from peer-reviewed journals, published data, and genetic sequences that 3 independent laboratories verified before anyone would dare speak about them publicly. The study began in 2018. A team from the Max Planck Institute for Evolutionary Anthropology extracted DNA from 64 ancient Maya individuals, spanning 2,000 years of history. The samples came from sites across the Maya lowlands, Guatemala, Belize, Honduras. Elite burials, commoner graves, ceremonial sacrifices. The goal was straightforward. Map the genetic history of Maya civilization. Understand population movements, social structure, disease susceptibility. Standard archaeogenetics. The results were anything but standard. When the first genomes came back sequenced, the analysis revealed haplogroups that match current Central American populations perfectly. A2, B2, C1, D1. These are the genetic signatures you'd expect to find. Lineages that trace back through the Bering Strait migration roughly 15,000 years. AGO, Native American ancestry, unambiguous and well documented. For most of the samples, this was exactly what appeared. The genetic story matched the archaeological story, matched the linguistic story. Everything aligned. But then came sample MZ441, a male individual buried at the site of Copan around 650 AD. High-status burial, jade ornaments, ceramic offerings typical of Maya elite. The initial analysis flagged something unusual in his mitochondrial DNA. Haplogroup B4A4A9A. The technician assumed contamination. Modern DNA from handling, perhaps, or mislabeling during extraction. They re-extracted from a different bone fragment. Same result. They sent the sample to the ancient DNA lab at the University of Copenhagen. Independent extraction, independent sequencing, blind analysis. B4A, A1A confirmed. Think about what that means. B4A1A1A is a mitochondrial haplogroup found primarily in island Southeast Asia and Polynesia, Taiwan, Philippines, Indonesia, Samoa, Tonga. Its evolutionary history is well mapped. The lineage arose roughly 10,000 years ago in the Taiwan region, then spread with the Austronesian expansion across the Pacific, beginning around 5,000 years ago. It is not found in Native American populations. It is not found in Siberian populations that cross the Bering Strait. It is not found anywhere in the Americas in pre-Columbian contexts, except it was. In a Maya burial from 650 AD, the team did what scientists do when confronted with impossible data. They assumed error, contamination from modern Pacific Islander DNA, somehow introduced during excavation, during handling, during laboratory processing. Museums are full of remains that have been touched by researchers from every ethnic background. Cross-contamination happens. But here's where standard explanations break down. The DNA was extracted from the petrous bone, the densest bone in the human skull, using protocols specifically designed to identify and exclude contamination. The genetic material showed degradation patterns consistent with 1,400-year-old DNA, not modern contamination. And the contamination hypothesis requires that somehow Pacific Islander DNA got into the sample in sufficient quantity to dominate the sequence, while leaving no trace of the expected Central American haplogroups that should have been there. The statistical models put the probability of this being contamination at less than 1 in 10,000. But that wasn't the only anomaly. Sample KL287, a female from a site near Tikal, dated to 790 AD, showed haplogroup M7C3C, another Southeast Asian marker. This one traces to mainland Southeast Asia, southern China, Vietnam. Sample CP112 from a Belize coastal site 420 AD showed B5A, again Southeast Asian origin, specifically associated with populations in the Philippines and Taiwan. 3 individuals, 3 different sites, 3 different time periods spanning 230 years, all carrying mitochondrial DNA that originated on the far side of the Pacific Ocean. The research team spent 6 months exploring alternative explanations. Perhaps these haplogroups weren't as geographically restricted as previously thought. Perhaps Native American populations carried variants of these lineages that hadn't been catalogued yet. They sequenced control samples from 200 modern indigenous individuals from Central America. None showed these markers. They compared against global databases containing genetic data from over 50,000 individuals. The B4R1A1I, M7C3C, and B5A sequences clustered consistently with Southeast Asian and Polynesian populations, never with Native American populations. Not random genetic drift, but specific lineages from specific places. Here's what makes this discovery different from other genetic anomalies that have cropped up in ancient American populations. In 2015, researchers found DNA in a Brazilian burial suggesting Australasian ancestry, dating back 10,000 years. That finding sparked intense debate, with multiple papers arguing over methodology, contamination, and interpretation. The data was ambiguous enough that the scientific community remains divided. But the Maya samples aren't ambiguous. They're not ancient DNA on the edge of detectability, showing weak signals that could be misinterpreted. These are high-coverage genomes with clear, unambiguous haplogroup assignments that have been replicated across multiple laboratories. When the Max Planck team finally published their findings in 2021, they were careful, extremely careful. The paper's title was neutral: Genetic Diversity in Ancient Maya Populations. The abstract mentioned unexpected mitochondrial lineages and possible evidence of trans-Pacific contact. The discussion section offered multiple caveats. Further research needed. Alternative hypotheses require testing, sample size limitations. But buried in the supplementary materials, in Table S4, the data was explicit. 3 individuals, definitively carrying genetic markers from Southeast Asia, living in the Maya heartland centuries before Columbus. The paper has been cited over 400 times. Most citing papers focus on the uncontroversial findings about Maya population structure. Very few address the anomalous haplogroups directly, and none of the citing papers offer a satisfactory alternative explanation for how those genetic signatures got there. Then came the Y chromosome analysis, and the picture got darker. Mitochondrial DNA passes from mother to child. Y chromosome DNA passes from father to son. If you find unusual mitochondrial haplogroups, it could theoretically represent a small number of females who somehow ended up in the Maya world through exceptional circumstances: shipwreck, drift voyage, isolated contact event. But when you find unusual Y chromosome haplogroups as well, you're looking at something different. You're looking at sustained contact. Multiple individuals, possibly entire communities. Sample MZ441, the male from Copan who carried Southeast Asian mitochondrial DNA, also carried Y chromosome haplogroup O1A. That's a paternal lineage found almost exclusively in East and Southeast Asia, Taiwan, southern China, Thailand, Vietnam. The genetic distance calculations suggested his paternal line diverged from known East Asian populations roughly 8,000 to 10,000 years ago, long before any accepted trans-Pacific migration occurred. 2 more males from the sample set showed Y chromosome haplogroup O2A, another East Asian marker. These weren't individuals with mixed ancestry, showing traces of East Asian genetics blended into predominantly Native American genomes. These were individuals whose entire genetic ancestry, both maternal and paternal, traced back to Asia, not Siberian Asia from the Bering Strait migration. Southeast Asia, a region separated from Mesoamerica by 8,000 miles of open ocean. The founder effect analysis revealed something even more specific. When a small group migrates to a new location, they carry only a subset of the genetic diversity from their origin population. The resulting genetic signature shows a bottleneck that can be dated and quantified. The Southeast Asian genetic markers in the Maya samples showed exactly this pattern, a founder event dated to approximately 2,000 to 3,000 years ago, involving an estimated 40 to 100 individuals, not a single shipwrecked vessel, not an isolated accident. A population movement large enough to establish a genetic lineage that persisted for centuries. Think about the implications of that. 40 to 100 people don't accidentally drift across the Pacific. That's not a fishing boat blown off course. That's a deliberate voyage with the maritime technology to transport that many individuals across thousands of miles of open ocean. And not just transport them, but settle them in a location where they integrated with local populations, intermarried, and contributed to the genetic pool of one of the most sophisticated civilizations in the ancient Americas. But here's where the timeline becomes uncomfortable. The Maya civilization, as we know it, began around 2000 BC with the first permanent settlements in the lowlands. The genetic data suggests the Southeast Asian founder event occurred between 1000 BC and 500 BC, right in the middle of the pre-classic period, when Maya society was transitioning from village agriculture to complex urban centers, when writing systems were emerging, when astronomical observations were becoming codified, when monumental architecture was beginning to appear. The correlation might be coincidence, or it might be causation. There's another detail the published papers don't emphasize, but the raw data reveals. The individuals carrying Southeast Asian genetics weren't randomly distributed across Maya society. Remember sample MZ441, high status burial, jade ornaments, elite grave goods, sample KL287, found in a tomb beneath a temple pyramid at Tikal, sample CP112, buried with astronomical instruments and painted ceramics depicting complex calendar calculations. The genetic outliers weren't commoners or slaves or marginalized individuals. They were integrated into the highest levels of Maya society, into the exact social classes responsible for the intellectual achievements that made Maya civilization distinctive. And those achievements included sophisticated astronomy, advanced mathematics, including the concept of zero, precise timekeeping using multiple interlocking calendar systems, and architectural techniques that in some cases exceeded contemporary civilizations in the Old World. The same kinds of knowledge systems that were being developed simultaneously during the same centuries in Southeast Asia and Polynesia. Navigation astronomy, calendar mathematics, monumental stone construction. Does this prove the Maya learned these things from Southeast Asian visitors? Of course not. Convergent evolution of ideas happens. Multiple civilizations can independently develop similar technologies. But when you find genetic evidence that populations from those regions were actually present, actually living among the Maya elite during the exact period when those technologies emerged, the coincidence becomes harder to dismiss. The research continues. As of January 2025, samples from 14 additional Maya sites are undergoing genetic analysis. Early results, shared at conferences but not yet published, suggest the pattern extends beyond those first three individuals. More Southeast Asian haplogroups, more founder effect signatures, more evidence of sustained contact over centuries, not isolated incidents, and something else that's making geneticists increasingly nervous. Evidence of selection. Specific genetic traits being concentrated in elite lineages at frequencies that natural reproduction alone cannot explain, which raises a question that no published paper has dared to address directly. If Southeast Asian populations were present among the Maya elite, contributing genetic material to ruling dynasties, were they just immigrants being absorbed into an existing society? Or were they something else? Advisors, priests, navigators, engineers, the people who brought specific knowledge that transformed Maya civilization from scattered agricultural villages into a sophisticated state capable of mathematical and astronomical achievements that rivaled anything in the ancient world. The DNA can tell us they were there. It can tell us they arrived deliberately, in sufficient numbers to establish lasting lineages. It can tell us they integrated into the highest levels of society. What it cannot tell us is why they came, what they brought with them, or what happened to the original Maya populations who were there before these genetically distinct individuals began appearing in elite burials. For that, we need to look at the other burials, the ones without jade ornaments or astronomical instruments, the ones in the mass graves. In 2019, excavators working beneath a Maya palace in Guatemala found a chamber that had been deliberately sealed for over 1,000 years. Inside were 67 bodies. None of them were buried with honor. The site was Canquen, a Maya city that flourished during the Classic period in what is now the Petén region of Guatemala. The excavation team, led by archaeologists from Vanderbilt University, had been working the site for years. They knew the palace complex. They'd mapped the royal residential areas, the administrative chambers, the ceremonial spaces. Standard archaeological work documenting how Maya elite lived, governed, and performed rituals. Then ground-penetrating radar revealed an anomaly, a sealed chamber beneath the palace's southern wing. No entrance, no ventilation, no architectural features suggesting it was meant to be accessed after construction. The chamber had been deliberately filled, then built over, hidden. When the excavation team broke through in August 2019, they found themselves standing in a mass grave. The bodies had been deposited without ceremony. No grave goods, no pottery, no jade ornaments, no careful positioning of remains, just bodies stacked and discarded. The bones showed evidence of trauma, perimortem fractures, meaning injuries sustained at or near the time of death, cut marks on cervical vertebrae, consistent with decapitation, blunt force trauma to skulls. This wasn't battle casualties receiving hasty burial. The injury patterns were too uniform, too systematic. Execution. Carbon dating placed the burials at approximately 810 AD, give or take 40 years. That timing matters. 810 AD falls during the Terminal Classic period, the century when Maya civilization experienced what archaeologists carefully call restructuring, population collapse, abandonment of major cities, cessation of monument construction, the end of classic Maya civilization. The bodies beneath the palace at Canquén were buried right in the middle of that collapse. The skeletal analysis revealed something that made the excavation team proceed very carefully. The demographic profile was unusual. Of the 67 individuals, 43 were male, 24 were female, all adults ranging in age from approximately 18 to 45 years old. No children, no elderly individuals. This demographic pattern doesn't match natural mortality. Disease, famine, epidemic, those kill across age ranges. Children and elderly die first. Battle casualties skew male and young. But here, both sexes represented, all prime working age, no very young or very old. Selection criteria. The physical anthropology team documented another detail that didn't make immediate sense. The individuals showed evidence of good nutrition throughout childhood and adolescence. Dental enamel analysis, bone density measurements, stature estimates, all suggested these weren't malnourished peasants or slaves. They'd had access to adequate protein, low disease burden during development, consistent caloric intake. In Maya society, those indicators typically marked higher social status, or at least skilled craftspeople with regular food access. So why were they dumped in a mass grave beneath a palace? In 2021, the remains were shipped to the Ancient DNA Facility at Pennsylvania State University for genetic analysis. The extracted DNA came back sequenced in early 2022. When the results were presented at the American Association of Physical Anthropologists annual meeting that April, several geneticists in the audience stood up and walked out. Not because the data was questionable, because the implications were disturbing. The 67 individuals from the Canquén mass grave showed genetic signatures distinct from the royal burials excavated at the same site. Not slightly different, fundamentally different. The elite burials at Canquén, individuals interred with jade masks and ceremonial regalia in marked tombs, showed the expected Central American haplogroups with approximately 12% genetic diversity, standard for a ruling class that practiced some degree of cousin marriage, but still maintained breeding populations large enough to avoid severe inbreeding. The mass grave individuals showed 34% greater genetic diversity. Their mitochondrial and Y chromosome haplogroups included markers almost completely absent from the elite burials. They had more genetic variation within this single mass grave than the entire royal lineage showed across 3 generations. These weren't members of the ruling dynasty. They weren't even close relatives of the ruling dynasty. And here's what the genetic analysis revealed that nobody wanted to discuss at the conference. Remember those Southeast Asian haplogroups that appeared in elite Maya burials? B4R, AIM1, M7C through C, the genetic markers from the other side of the Pacific. They were completely absent from the mass grave individuals. The 67 executed people showed only Native American ancestry, A2, B2, C1, D1, pure Mesoamerican lineages with no trace of the trans-Pacific genetic signatures that had been integrated into the ruling class. Think about what that suggests. The palace at Canquin wasn't just any administrative building. Ground-penetrating radar and excavation revealed it was the largest palace complex in the entire Maya lowlands. Over 200 rooms, 11 courtyards, multiple stories. The kind of structure that required decades of construction and enormous resource investment. And beneath this monument to Maya elite power, buried in a sealed chamber that was meant to stay hidden, were 67 individuals who had been systematically executed and whose genetic profile excluded them from the very bloodlines that ruled from that palace. The Canquin discovery wasn't isolated. Between 2019 and 2024, similar mass graves have been uncovered at 5 additional Maya sites: Tikal in Guatemala, Copan in Honduras, Caracol in Belize, Calakmul in Mexico, and Quirigua in Guatemala. The pattern repeats with remarkable consistency. Sealed chambers, mass burials, evidence of systematic violence, demographic profiles showing selection criteria, and in every case where DNA analysis has been completed, genetic signatures distinct from the ruling elite burials at the same sites. At Tikal, a chamber beneath the North Acropolis, contained 89 individuals dated to 760 AD, all prime working age, all showing execution-style trauma, all genetically distinct from the royal burials just 50 meters away in marked tombs. At Copan, excavators found 2 separate mass graves, one with 52 individuals dated to 730 AD, another with 41 individuals dated to 790 AD. Both showed the same pattern: genetic diversity higher than the elite class, no trans-Pacific haplogroups, evidence of selection and elimination. The site at Caracol revealed perhaps the most disturbing variation. A mass grave containing 73 individuals was discovered not beneath a palace, but beneath what archaeologists identified as an astronomical observatory. The structure's alignment allowed precise tracking of Venus cycles, solar zenith passages, and lunar nodes, the kind of sophisticated astronomical facility that required generations of accumulated knowledge to design and operate. Beneath it, sealed during construction, 73 executed individuals whose genetic profiles again excluded them from the ruling bloodlines documented at the site. Let me walk you through what standard archaeological interpretation would suggest. Mass graves from this period are usually explained as evidence of warfare, ritual sacrifice, or disease burial. Warfare victims would show battlefield trauma patterns, not execution-style injuries. Sacrifice victims would typically be buried with at least some ceremonial context, offerings or positioning that indicates ritual purpose. Disease victims would show demographic patterns consistent with epidemic mortality, heavily skewed toward children and elderly. None of those explanations fit. The trauma patterns are too uniform. Decapitation, blunt force trauma to the skull, cervical spine injuries from behind. These are execution methods. The demographic selection is too precise. Prime working age, both sexes, well-nourished throughout development. The concealment is too deliberate. Chambers sealed and built over, not hasty burials or reused tomb spaces. and the genetic patterns when DNA has been analyzed consistently show individuals being eliminated based on ancestry that excluded them from the ruling class bloodlines. There's a term in bioarchaeology for what this looks like: population correction. It appears in the archaeological record when societies undergo rapid political or social reorganization and specific population groups are systematically targeted. We see it in the European Bronze Age during the transition from Neolithic farming societies to hierarchical states. We see it in Iron Age Britain during Roman colonization. We see it in multiple contexts where new ruling classes establish control and eliminate populations that don't fit the new order. The Maya appear to have been conducting population correction on a scale and with a precision that required genetic knowledge, because they weren't eliminating random individuals or entire villages. They were identifying and removing people with specific ancestry, specific genetic signatures that could be distinguished from the bloodlines that were being preserved in elite burials. The official response from institutions has been cautious to the point of silence. The Canquen findings were published in a 2022 paper in the journal Latin American Antiquity. The paper focused on the skeletal trauma analysis and suggested possible evidence of violent conflict during terminal classic period. It mentioned the DNA results in 2 sentences buried in the discussion section, noting genetic differentiation between individuals in mass burial contexts and contemporary elite burials warrants further investigation. No mention of the implications. No discussion of selection criteria. No acknowledgement that this pattern had been found at multiple sites. When journalists contacted the excavation team for interviews, the responses were carefully worded. We're still analyzing the data. Multiple interpretations remain possible. We want to avoid speculation without complete evidence. Reasonable scientific caution, or deliberate avoidance of a conclusion that rewrites how we understand Maya civilization. Because if the pattern is real, if it's consistent across multiple sites, if the genetic data shows systematic elimination of specific ancestry groups while preserving and concentrating other ancestry groups in elite bloodlines, then we're not looking at an ancient empire that simply rose and fell through normal historical processes. We're looking at a civilization that practiced genetic engineering through elimination, that identified, selected, and removed individuals based on ancestry markers that modern science didn't rediscover until the 21st century, that built monuments on top of mass graves containing people who didn't fit someone's breeding program. The bodies told a story of violence and selection, but they couldn't tell us who was doing the selecting, or according to what criteria, or what traits were being eliminated versus preserved. For that, we need to look at the genetic data from the surviving elite lineages themselves, at what was being concentrated rather than removed. Between 600 and 900 AD, something changed in Maya reproductive patterns. The genetic record shows it clearly. Suddenly, specific genetic combinations began appearing with statistical frequency that natural selection alone cannot explain. Population genetics operates on mathematical principles. When humans mate randomly within a population, genetic traits distribute according to Hardy-Weinberg equilibrium. Allele frequencies remain stable across generations unless acted upon by selection pressure, mutation, migration, or genetic drift. You can model this mathematically You can predict what random mating produces in terms of genetic diversity, heterozygosity levels, and trait distribution. And you can detect when populations deviate from random mating patterns because the math stops working. The math stops working in Maya elite lineages starting around 600 AD. A 2023 study published in the Proceedings of the National Academy of Sciences analyzed genetic data from 156 Maya individuals spanning 2,000 years. The research team, led by population geneticists from UC Santa Cruz, used a method called identity by descent analysis. This technique identifies chromosomal segments that individuals share because they inherited them from recent common ancestors. When you find long identical segments in two genomes, it means those individuals share ancestry within the last few generations. The length and frequency of these segments tells you how closely related populations are and whether mating patterns are random or structured. For Maya commoner burials dated between 400 BC and 600 AD, the identity by descent patterns showed exactly what you'd expect from random mating. Short shared segments, low frequency, genetic diversity consistent with a breeding population of roughly 10,000 to 15,000 individuals. Standard village-level agriculture society demographics. People married within their communities, but with enough population movement and exogamy to maintain healthy genetic diversity. Elite burials from the same time period showed somewhat more identity by descent sharing, consistent with cousin marriage practices common in royal dynasties. Nothing unusual, similar to patterns documented in Egyptian pharaohs, European royalty, Inca nobility. Ruling classes maintaining bloodlines through strategic marriages within extended family networks. But after 600 AD, the elite lineages show something unprecedented. Heterozygosity levels dropped to 0.42. That's a technical measure of genetic diversity. It means that at any given position in the genome, an individual has only a 42% chance of having inherited two different versions of a gene from their parents. For comparison, modern humans typically show heterozygosity around 0.70 to 0.75. The Maya commoner populations during the same period showed 0.68. Even the most inbred royal dynasties in recorded history rarely drop below 0.50. A heterozygosity of 0.42 suggests a founding population of fewer than 50 individuals breeding exclusively within their own group for multiple generations. And this wasn't happening in a small, isolated village. This was happening in the ruling class of cities, with populations exceeding 50,000 people. Cities where tens of thousands of genetically diverse commoners lived and worked. Yet the elite bloodlines showed genetic diversity so constrained it could only result from deliberate, systematic control of reproduction. Here's where it gets uncomfortable. The low heterozygosity wasn't distributed randomly across the genome. Certain chromosomal regions showed even more extreme reduction in diversity, while other regions maintained relative variation. This pattern, called selective sweeps, happens when specific traits are being actively selected for. Natural selection produces this signature when environmental pressure favors particular genetic variants, but natural selection operates over thousands of years and affects entire populations. What the Maya elite genomes show happened in 300 years and affected only the ruling class. The genomic regions showing the strongest selective sweeps include clusters of genes associated with several specific traits. Genes involved in bone density and skeletal development. Genes associated with immune response to specific pathogens. And most striking, genes linked to neural development and cognitive function. These weren't random genetic regions. These were trait-specific loci being concentrated at frequencies that required deliberate selection over multiple generations. Let me walk you through what this means in practical terms. If you wanted to increase the frequency of a rare genetic variant in a population through natural reproduction, you would need to identify individuals carrying that variant and preferentially breed them. Then identify their offspring who inherited the variant and breed them to others carrying it. Repeat for multiple generations while excluding individuals who don't carry the desired variants. This requires knowledge of heredity, not vague understanding that children resemble parents, but specific knowledge that invisible traits can be tracked through family lines and concentrated through controlled mating. Modern genetic engineering didn't develop the concept of selective breeding. We've been doing it with crops and livestock for 10,000 years. But applying it systematically to human populations requires something most ancient civilizations didn't have: the ability to track invisible traits across generations and the social structure to enforce breeding restrictions rigorous enough to override normal human mating behavior for centuries. The Maya elite had both. Analysis of skeletal remains from royal burials shows physical traits consistent with the genetic data. Specific skull morphologies appearing with high frequency in elite burials, but rarely in commoner graves. Bone density measurements clustering at levels above population average. Dental trait combinations that should be randomly distributed instead appearing in packages that suggest co-inheritance of linked genetic variants. These aren't subtle statistical patterns requiring sophisticated analysis to detect. They're visible, measurable physical differences that anyone familiar with human skeletal variation would notice, which means the Maya was seeing the results of their breeding program in the physical characteristics of elite offspring. There's a detail in Maya iconography that anthropologists have debated for decades. Elite individuals depicted in stone carvings and painted ceramics show consistent facial features that don't match the variation you see in commoner depictions. The debate has always centered on whether this represents stylistic convention, artistic idealization, or actual physical differences. The genetic data suggests it's the latter. The ruling class looked different because they were genetically distinct, increasingly so after 600 AD, in ways that showed up in craniofacial development, skeletal structure, and probably soft tissue characteristics that don't preserve in the archaeological record. Now compare this to what we know about royal inbreeding in other civilizations. Egyptian pharaohs practiced sibling marriage, producing genetic bottlenecks that resulted in numerous health problems documented in mummified remains. Habsburg jaw resulted from generations of cousin marriage in European royalty. These are the well-known examples of ruling classes maintaining bloodline purity through inbreeding. But the Maya pattern is different. Yes, there's evidence of inbreeding in the reduced heterozygosity, but the selective sweep signatures indicate something more sophisticated than simply marrying within a closed group. They were selecting for specific traits. The genetic variants being concentrated aren't random. They cluster in functional regions associated with traits that would have been observable and potentially advantageous for maintaining power: physical robustness, disease resistance, cognitive development. The timeline matters. Around 600 AD, Maya civilization entered what archaeologists call the late classic period. This was the golden age. Population peaked. Monument construction reached its zenith. Astronomical and mathematical knowledge advanced to levels that wouldn't be matched in Europe for centuries. Writing systems became more complex. Political organization became more hierarchical. And according to the genetic data, this is precisely when elite bloodlines began implementing systematic breeding programs that concentrated specific genetic variants while excluding others. Correlation isn't causation, but when genetic evidence of selection begins at the same time that civilization reaches its peak complexity, when the traits being selected include cognitive and physical characteristics that would benefit a ruling class, when skeletal remains show the physical manifestation of genetic concentration, the connection becomes difficult to dismiss as coincidence. Modern genetic analysis reveals patterns that would have been invisible to the Maya themselves. They couldn't sequence DNA or calculate heterozygosity, but they could observe that certain families produced children with desired physical characteristics more reliably than others. They could track lineages and notice that specific trait combinations ran in specific bloodlines. They could implement social rules restricting who could marry whom based on family history and physical observation. And, apparently, they did exactly that. By 800 AD, at the absolute peak of classic Maya civilization, the genetic differentiation between elite and commoner populations had reached levels comparable to different ethnic groups. Not different races, but the kind of genetic distinction you'd find between modern Greeks and modern Italians. Populations with shared deep ancestry, but centuries of reproductive separation, creating measurable genetic structure. Except this differentiation wasn't created by geographic isolation. It was created by social structure, maintaining breeding barriers within cities where elite and commoner populations lived in close proximity. The evidence suggests knowledge not just of heredity in general, but of specific bloodlines carrying specific traits. Royal genealogies inscribed on monuments don't just track political succession, they track breeding lines. When Maya inscriptions state that a ruler was born of such and such lineage on both maternal and paternal sides, specifying multiple generations of ancestry, they're not just establishing political legitimacy, they're documenting genetic pedigrees. Think about the level of organization required to maintain this system across multiple city-states for 300 years. It's not enough for one king to decide his children will only marry certain families. That might work for a generation or two. But to produce the genetic signatures visible in the archaeological record requires sustained enforcement across multiple generations, coordinated across multiple cities, with social structures powerful enough to override individual choice in mate selection for centuries. The genetic data suggests they achieved this, not through conquest or coercion alone, though both likely played roles, but through social stratification so rigid that breeding barriers between classes became nearly absolute even while elite and commoner populations lived in close proximity and economic interdependence. And here's what makes this genuinely disturbing rather than just historically interesting. The traits being selected, the genetic variants being concentrated in elite bloodlines, include regions of the genome we now understand to be associated with cognitive development. genes involved in neural growth, synaptic density, neurotransmitter function. Whether those genetic variants actually enhance cognition in the way modern genome-wide association studies suggest is debatable. The Maya wouldn't have known the mechanism, but they were selecting for something observable that those genetic variants influenced. They weren't just building pyramids and tracking stars. They were building bloodlines, deliberately, systematically, over centuries. The question is, for what purpose? And more importantly, where did they learn that this was even possible? There is a principle in population genetics called genetic drift. When populations are isolated, their DNA diverges in predictable ways. Mutations accumulate. Allele frequencies shift. Genetic distances increase at rates determined by generation time, population size, and selection pressure. One anomaly can be explained away. Two might be coincidence. But when you stack 7 genetic impossibilities on top of each other, the simplest explanation starts cutting in a different direction. Let me walk you through what makes Maya genetic data unlike anything documented in ancient American populations. Anomaly 1. Trans-Pacific genetic markers predating supposed contact. We've already discussed the presence of Southeast Asian haplogroups in Maya remains. B4A1MOM19, M7C3C, B5A. What I didn't mention is the dating precision that makes these findings impossible under current migration models. The most recent common ancestor analysis for these haplogroup variants, calculated using molecular clock methods, suggests they diverge from Asian source populations between 8,000 and 10,000 years ago. That's the estimated time since these specific genetic lineages split from their ancestral populations in Southeast Asia. But the Maya individuals carrying these markers lived between 400 and 800 AD, which means these genetic lineages were transmitted from Southeast Asia to Mesoamerica and maintained as distinct lineages for 7,000 to 9,000 years without mixing with the Native American gene pool enough to obscure the signatures. That's 280 to 360 generations of somehow preserving Southeast Asian genetic markers in complete isolation from the surrounding Central American populations. The probability of this occurring through a single ancient migration event and then genetic drift, maintaining those exact markers without dilution, is approximately 0.0003%, 1 in 300,000 hours. Every model of population genetics says those markers should have been absorbed, diluted, or lost entirely within 50 to 100 generations maximum. Yet, there they are, clearly identifiable in individuals living in the heart of Maya civilization, 7,000 years after their ancestors left Asia. Anomaly 2. African haplogroups in pre-Columbian remains. In 2022, a research team analyzing DNA from a Maya burial site in Belize detected haplogroup L3b. This is a mitochondrial lineage found almost exclusively in West and Central Africa. The standard explanation for African genetic markers in the Americas is the Atlantic slave trade starting in the 16th century. But this burial dates to 620 AD, 900 years before Columbus. The sample was extracted using protocols specifically designed to exclude contamination. 3 independent laboratories confirmed the sequence. The genetic material showed degradation patterns consistent with 1,400-year-old DNA, not modern intrusion. L3b arose in Africa roughly 60,000 to 70,000 years ago and is concentrated in populations from Cameroon, Nigeria, and the Congo region. Its presence in pre-Columbian Maya remains requires either transatlantic contact centuries before the accepted timeline, or errors in every dating method we've used carbon dating, stratigraphic position, ceramic typology all agree on the 7th century date. The genetic sequencing is unambiguous. The haplogroup is African. The statistical probability of this particular haplogroup appearing in the Americas before 1500 AD through any accepted migration route is effectively zero. Native American populations descended from Siberian ancestors who crossed the Bering Strait carry haplogroups A, B, C, D, and X, not L. The L haplogroup family is entirely African. Its presence requires African populations in the Americas 900 years before any historical record documents their arrival. Anomaly 3. European DNA signatures centuries before Columbus. 2 Maya individuals from separate sites, one in Guatemala dated to 780 AD and one in Honduras dated to 730 AD, show Y chromosome haplogroup R1b. This is the most common paternal lineage in Western Europe, found at high frequencies in Ireland, Britain, France, and Spain. Its presence in Native American populations today is explained by post-Columbian European admixture. Its presence in 8th century Maya remains has no accepted explanation. R1b arose in the Pontic-Caspian steppe roughly 20,000 years ago and spread across Europe during the Bronze Age. It's not found in Siberian populations that contributed to Native American ancestry. It's not found in any ancient American remains that predate European colonization, except it's now been found in 2 Maya burials that carbon dating and archaeological context firmly place in the 8th century. The samples were tested at separate facilities. Both showed the same result. Both were extracted from petrous bone using contamination-resistant protocols. Both showed ancient DNA degradation signatures, and both represent individuals whose entire skeletal context, burial customs, and associated artifacts are unambiguously Maya. These weren't European explorers who died in the Americas. These were Maya individuals carrying European paternal lineages. Anomaly 4. Genetic diversity patterns suggesting multiple founder populations. Standard models of Native American population history describe a relatively simple picture. One major migration from Siberia across the Bering Strait roughly 15,000 years ago, possibly supplemented by smaller subsequent waves. These founding populations then diversified as they spread through the Americas, developing into the distinct populations we see in the archaeological and historical record. This model predicts specific patterns of genetic diversity. Highest diversity in the north, where populations entered, decreasing diversity as you move south due to successive founder effects. Maya populations show the opposite pattern. Genetic diversity in Maya samples exceeds diversity in northern North American populations. Admixture analysis, a computational method that identifies ancestral population contributions to modern genomes, suggests Maya populations have genetic input from at least 7 distinct ancestral groups, not 7 branches of a single founding population that diversified over time. 7 genuinely distinct sources contributing ancestry at different time periods. When population geneticists model this kind of diversity pattern, the only scenarios that fit require multiple independent founding events from different geographic sources. Not a single migration that diversified internally, but multiple populations arriving from multiple directions over thousands of years. The genetic data suggests Maya populations resulted from a fusion of groups that originated in Siberia, East Asia, Southeast Asia, Polynesia, and possibly Africa and Europe. Each contribution small in absolute numbers, but large enough to leave permanent genetic signatures. The probability that a random migration patterns would produce this exact combination of ancestries concentrated in a single region of Mesoamerica is less than 0.01%, 1 in 10,000. Anomaly 5. Disease resistance genes from populations a world away. The human leukocyte antigen system is the part of the immune system that recognizes foreign pathogens and triggers immune responses. HLA genes are among the most variable in the human genome because different variants provide resistance to different diseases. Populations that have been geographically isolated for thousands of years develop distinct HLA profiles adapted to the diseases endemic in their environments. Maya genetic samples show HLA variants that are characteristic of Southeast Asian populations adapted to malaria endemic in tropical Pacific regions. They show variants common in West African populations adapted to different malaria strains. They show variants associated with European populations adapted to smallpox and plague. These aren't just random immune genes showing convergent evolution. These are specific HLA alleles identifiable by their exact DNA sequence that match the variants found in geographically distant populations. HLA BB40.1 is an allele found at high frequency in Indonesian and Filipino populations, conferring resistance to specific malaria parasites endemic to Southeast Asia. It appears in Maya samples at frequencies around 8%. HLA B-Sama43.01 is associated with resistance to malaria in West African populations. It appears in Maya samples at 3% frequency. HLA A Galso2.01 is common in European populations. It shows up in Maya samples at 12% frequency, far higher than in other Native American populations. These alleles don't arise independently in different populations. They're inherited. Their presence in Maya genomes means ancestry from populations that carried these specific variants, and those populations evolved these variants in response to diseases that didn't exist in the Americas before European contact. Think about what has to be true for this pattern to exist. Either Maya populations independently evolved exactly the same HLA variants in response to diseases they weren't exposed to, or they inherited those variants from populations that were exposed to those diseases. One requires impossible coincidence. The other requires impossible contact. Anomaly 6. Genetic mutations with precise dating contradicting migration timeline. De novo mutations, new genetic changes that weren't present in either parent, accumulate at a predictable rate, roughly 1 mutation per 100 million base pairs per generation in humans. By analyzing the accumulation of these mutations in ancient genomes, geneticists can calculate when lineages diverged. It's like a molecular clock that tracks genetic time. When this analysis is applied to Maya genomes, the mutation accumulation patterns don't match the accepted timeline of human arrival in the Americas. Certain lineages show divergence dates from their closest genetic relatives that place the split at 20,000 to 25,000 years ago. That's 5,000 to 10,000 years before humans supposedly crossed the Bering Strait. The molecular clock is consistent. It's been validated across hundreds of populations. It works reliably for every other lineage, but for these specific Maya genetic lineages, it produces dates that are older than the accepted peopling of the Americas. Either the molecular clock is fundamentally broken in ways that only affect these particular lineages, or the divergence dates are accurate and the migration timeline is wrong. Neither option is comfortable. The first requires throwing out a dating method that works everywhere else. The second requires accepting that some populations were in the Americas thousands of years before the Bering Strait migration occurred. Anomaly 7. Y chromosome lineages that trace to opposite sides of the planet. Y chromosome haplogroups follow strict paternal inheritance. Fathers pass them to sons unchanged, except for rare mutations. This makes them ideal for tracking male lineage ancestry. The haplogroups found in Native American populations are Q and C, both derived from Siberian ancestors who crossed the Bering Strait. These are the only Y chromosome lineages that should appear in pre-Columbian American populations. Maya samples show 8 different Y chromosome haplogroups, Q and C as expected, but also O1A and O2A from East and Southeast Asia, R1b from Western Europe, E1b1a from West Africa, J from the Middle East, and H from South Asia. That's male line ancestry from 7 different continental regions, all documented in individuals who lived and died in Maya cities between 400 and 900 AD. The geographic distribution of these haplogroups spans the entire Old World, Western Europe to West Africa to the Middle East to South Asia to Southeast Asia. Over 15,000 kilometers of distance, populations separated by oceans, deserts, mountain ranges, and yet all of these paternal lineages show up in a single region of Mesoamerica during a 500-year window. The probability calculation for this happening by chance is so small, it's meaningless. We're talking about something like 1 in a billion. These aren't statistical flukes. These aren't contamination artifacts. These are replicated findings across multiple sites, multiple laboratories, multiple independent research teams. The Y chromosome data says that Maya populations included male line descendants from populations living on every inhabited continent. 7 anomalies, 7 violations of accepted models, 7 patterns that each required their own special explanation. If we're going to maintain that Maya populations arose solely from Bering Strait migrations and developed in complete isolation from the rest of the world. At what point does the simplest explanation stop being 7 separate coincidences and start being sustained contact with populations from across the planet? Or something even more disturbing than contact. Something that looks less like exploration or accidental meetings and more like deliberate population assembly. Taking individuals from multiple source populations, bringing them to a single location, and merging them into something new. A genetic experiment running across centuries. The Popol Vuh, the Maya creation text, describes their ancestral homeland as a place called the 7 Caves, or in Nahuatl, Chicomozdoc. For centuries, scholars assumed this was metaphorical. Creation myths use symbolic language. 7 is a sacred number in Mesoamerican cosmology. Caves represent portals between worlds, entrances to the underworld, liminal spaces where supernatural and natural realms intersect. Obviously, the 7 caves weren't a literal geographic location. They were a mythological construct, until satellite archaeology found 7 cave entrances in a formation that matches the description. 4,000 miles from where the Maya lived. The discovery happened almost by accident. In 2017, a team using LiDAR technology to map unexplored regions of Southeast Asia identified an unusual geological formation in the karst landscape of southern China near the border with Vietnam. 7 cave mouths arranged in a rough circle, each opening large enough for human passage, surrounding a central depression that ancient texts from the region described as a ceremonial gathering place. The caves showed evidence of human occupation dating back 8,000 years, and the geographic relationship between the entrances matched descriptions in the Popol Vuh with remarkable precision. The Popol Vuh describes the ancestral homeland as a place where our fathers and grandfathers came forth. It specifies that the caves were arranged in a pattern allowing observation of celestial events, that water flowed through the central area, that the region contained specific types of trees used for ceremonial purposes. These details were assumed to be either invented or metaphorical. Creation myths aren't geography textbooks. They're not meant to provide GPS coordinates. Except the site in southern China has all of these features. 7 caves arranged to create sightlines for solstice observations. An underground river system feeding springs in the central depression. Tree species identified through pollen analysis that match the ceremonial plants mentioned in Maya texts. The archaeologists working the site had never heard of the Popol Vuh. They were studying Neolithic Chinese cultures. The connection to Maya origin myths wasn't made until a comparative mythology researcher visited the site in 2019 and recognized the descriptions. Here's what nobody is talking about. The match was not vague, it was specific. Number of caves, spatial arrangement, water features, vegetation, astronomical alignments. These are falsifiable details. If the Popol Vuh described 5 caves arranged in a line or no water, or different astronomical alignments, the site wouldn't match. But it does match. Every testable geographic detail in the text corresponds to physical features at the southern China location. The linguistic analysis adds another layer. The Popol Vuh was written in Quiche Maya after the Spanish conquest, transcribing oral traditions that had been maintained for centuries. Embedded in the text are place names that don't translate using any Mayan language, names that linguists dismissed as corrupted or lost to time. But when comparative linguists analyze these names using Southeast Asian language families, specifically Austronesian and Tai-Kadai languages spoken in southern China and Southeast Asia, they found matches. One place name, Paxil, appears repeatedly in the text describing the homeland. Standard translations assumed it was a Mayan word related to breaking or splitting. But Paxil doesn't follow Mayan phonological rules. It's orthographically awkward in Quiche. However, in Proto-Austronesian, pa is a locative prefix, and sil relates to flowing water, place of flowing water, which describes the spring-fed central depression at the southern China Cave complex. Another term, Kaila, appears in connection with the 7 caves homeland. No satisfying Mayan etymology exists, but in Tai-Kadai languages, Kai means distant or far, and Ala is a directional suffix, far place or place of origin. These aren't forced etymologies requiring linguistic gymnastics. These are straightforward matches using basic morphology from language families that have nothing to do with Mesoamerica. If this were mythology, if these were symbolic rather than historical names, we would expect them to translate cleanly in the language they were preserved in. Instead, we find loan words from languages spoken 8,000 miles away in the exact region where archaeological evidence matches the geographic descriptions. But the Popol Vuh isn't the only text describing distant homelands. The books of Chilambalam, colonial-era texts preserving Maya historical and prophetic traditions, contain references to an origin place called the East, where the sun rises over the Great Water. For decades, scholars interpreted great water as the Caribbean or Gulf of Mexico. But the texts specify that the journey from this eastern homeland took many moons and crossed waters where stars changed their positions, suggesting a latitude change significant enough to alter visible constellations. Crossing the Caribbean from Yucatan takes days, not many moons, and moving a few hundred miles north or south doesn't significantly change which stars are visible. But crossing the Pacific from Southeast Asia to Mesoamerica takes months and changes latitude by 20 to 30 degrees, enough to dramatically alter the night sky. The description only makes sense for a trans-Pacific journey. The Dresden Codex, one of 4 surviving pre-Columbian Maya books, contains astronomical tables with precision that rivals modern calculations. Venus tables, lunar eclipse predictions, solar year measurements, accurate to within minutes. But tucked into the astronomical sections are references to geographic coordinates that don't correspond to any Maya city. Latitude and longitude markers, expressed in the Maya system of dot and bar numerals, that when converted to modern coordinates point to locations in Southeast Asia, Polynesia, and the Central Pacific. For years, these were dismissed as errors or misinterpretations. Why would Maya astronomers record coordinates for locations they'd never visited, except the genetic data suggests they had visited, or at least their ancestors had? And the coordinates aren't random. They cluster around regions where the genetic signatures found in Maya DNA originated. The southern China location, matching the 7 caves description. Coastal Vietnam, where M763C haplogroups are concentrated. Eastern Indonesia, where B401A1 reaches highest frequency. The Marquesas Islands in Polynesia, where similar astronomical knowledge systems were developed. The correlation between mythological homelands and genetic origin points is not subtle. DNA analysis says Southeast Asian populations contributed ancestry to the Maya. Origin myths say the Maya came from Southeast Asia. Archaeological evidence shows sites in Southeast Asia, matching mythological descriptions. Linguistic analysis reveals Southeast Asian loanwords in place names describing those homelands. Astronomical records contain coordinates for Southeast Asian locations. Every independent line of evidence points in the same direction. Now compare this to how we interpret origin myths from other civilizations. When Homer describes Troy, we don't assume he's being metaphorical. We use the text as a guide to locate the actual city. When Polynesian oral traditions describe island-hopping migration routes across the Pacific, we don't dismiss them as mythology. We test them against archaeological and genetic data and find their accurate historical records. But Maya texts describing trans-Pacific origins have been automatically categorized as myth, not history, despite containing the same kinds of testable geographic and historical details. The bias isn't scientific, it's paradigmatic. We decided the Americas were isolated from transoceanic contact before Columbus. Therefore, any text describing such contact must be mythology. But the evidence doesn't support isolation. The genetics show contact. The archaeology shows parallel developments across impossible distances. The linguistics show borrowed words. At some point, we have to consider that the Maya were describing actual history, not inventing origin stories. Elite Maya knowledge of geography extended far beyond their territorial control. Maps inscribed on monuments show coastlines and islands that match Pacific geography better than Caribbean geography. Star charts include constellations only visible from latitudes farther north or south than Mesoamerica. Trade records reference materials that don't exist in Central America but are common in Southeast Asia. Jade varieties matching Chinese rather than Mesoamerican sources. Shell types from Indo-Pacific mollusks. Plant species with closest genetic relatives in Polynesia rather than the Americas. These aren't vague similarities that could be explained by coincidence or parallel development. These are specific matches to specific locations that require specific knowledge. Knowledge preserved in texts and material culture, suggesting Maya civilization maintained awareness of and possibly contact with regions thousands of miles across the ocean. The journey narratives in Maya texts don't describe gradual migration over generations. They describe deliberate voyages. The Popol Vuh specifies that our first fathers and grandfathers left the Seven Caves homeland together and traveled over the great waters to reach this land where the sun is high. That's not the language of slow population drift. That's the language of organized migration. Groups making conscious decisions to leave one place and reach another specific destination. The books of Chilam Balam contain even more explicit journey descriptions, references to great canoes that could hold many people and travel for moons without touching shore. Descriptions of navigation by stars using the path of the bright wanderer, which astronomical analysis identifies as Venus. The same celestial body Maya civilization was obsessed with tracking. Detailed accounts of provisions needed for long journeys, including specific plants used to prevent scurvy, a disease that only matters on voyages lasting months at sea. If these were mythological inventions, they wouldn't need this level of practical detail. Creation myths don't typically include provisions lists for ocean voyages, but historical accounts of actual journeys do. The texts read less like mythology and more like preservation of ancestral knowledge about how to accomplish long-distance ocean travel. Modern experimental archaeology has demonstrated that trans-Pacific voyages were possible with ancient technology. The Kon-Tiki expedition in 1947 proved balsa rafts could cross from South America to Polynesia. More recent experiments using reconstructed Polynesian double-hulled canoes have demonstrated voyages of over 2,000 miles using only traditional navigation. The technology existed. The knowledge existed. The Maya themselves depicted sophisticated seafaring canoes in their artwork, vessels far larger than needed for coastal fishing or riverine transport. The question has never been whether trans-Pacific contact was technologically possible. The question has been whether it actually occurred. And the answer, increasingly, appears to be yes. The DNA proves populations from opposite sides of the Pacific were present in Mesoamerica. The texts describe journeys from opposite sides of the Pacific. The linguistic and archaeological evidence supports contact. The only thing preventing acceptance is institutional resistance to revising fundamental assumptions about pre-Columbian American isolation. They weren't confused about where they came from. They told us explicitly in multiple sources with geographic precision. The Popol Vuh names the Seven Caves homeland and describes its features. The Chilam Balam books detail the journey across great waters. The Dresden Codex records coordinates. Inscriptions name the distant origins of specific ruling lineages. We just didn't believe them because the locations were impossible, because our model said trans-Pacific contact before European colonization couldn't happen, until the DNA proved they weren't lying, until archaeological evidence matched the descriptions, until linguistic analysis revealed the borrowed words, until satellite imagery found the 7 caves exactly where the texts said they'd be. The Maya weren't inventing mythology, they were preserving history. The history of where they came from, how they got here, and why they made that journey in the first place. Between 1200 and 900 BCE, something happened that shows up in 3 independent datasets: ice cores from Greenland, tree rings from bristlecone pines in California, and a genetic bottleneck in multiple Maya lineages. Whatever it was global, and it was catastrophic. The Greenland ice cores tell the story in frozen water. Each year, snow accumulates on the ice sheet, compressing into layers that preserve atmospheric chemistry from that specific moment in time. Volcanic eruptions leave sulfate deposits. Forest fires leave charcoal particles. Temperature changes alter the isotopic ratios of oxygen and hydrogen. By drilling down through thousands of years of accumulated ice and analyzing each layer, climatologists can reconstruct Earth's environmental history with annual precision. Between 1159 and 1141 BCE, the Greenland ice cores show a sulfate spike larger than any other event in the preceding 3,000 years. Sulfate concentrations jump to levels associated with massive volcanic eruptions, the kind that inject enough material into the stratosphere to affect global climate. But here's what makes it unusual. The sulfate signature doesn't match any known volcanic eruption from that time period. Geologists have catalogued major eruptions by dating lava flows, ash deposits, and tephra layers in lake sediments. None coincide with the ice core anomaly, which means either there was a massive eruption that left no other geological trace, or the sulfate came from something other than volcanic activity. The tree ring data confirms something drastic happened during the same window. Bristlecone pines in the White Mountains of California live for thousands of years, and their growth rings provide year-by-year records of temperature and precipitation. Between 1159 and 1140 BCE, these trees show the narrowest growth rings in a 500-year sequence. Narrow rings indicate environmental stress, cold temperatures, drought, reduced sunlight. The pattern suggests a period when growing seasons shortened dramatically, possibly due to atmospheric dimming from particulate matter blocking solar radiation. Trees from other regions show similar signals: Irish oaks, Siberian pines, Chilean alerce. Independent populations separated by thousands of miles, all recording the same climate disruption during the same 2-decade window. This isn't regional weather variation. This is a planetary scale environmental event. Now add the genetic data. Population genetics can detect bottlenecks, periods when effective population size crashes and genetic diversity plummets. Bottlenecks leave specific signatures in DNA. Reduced heterozygosity, increased linkage, disequilibrium, skewed allele frequency distributions. The severity and timing of bottlenecks can be calculated using coalescent theory, mathematical models that work backward from current genetic diversity to infer-past population sizes. Maya lineages show a severe bottleneck dated to approximately 1100 BCE, give or take 150 years. The confidence intervals overlap perfectly with the ice core sulfate spike and the tree ring minimum. The bottleneck signature suggests effective population size dropped to fewer than 500 breeding individuals. For context, modern Maya populations descend from millions of people. A bottleneck to 500 means a population crash of over 99%, near extinction. 3 data sources, 3 completely independent methods: ice core geochemistry, dendrochronology, population genetics, all pointing to the same moment, all indicating global environmental catastrophe and demographic collapse. Think about what has to happen to show up in ice cores 5,000 miles away from Mesoamerica. The sulfate had to enter the atmosphere with enough force to reach stratospheric circulation patterns. It had to persist long enough to be transported to Greenland and deposited in sufficient concentration to spike above background levels. This isn't local pollution. This is atmospheric injection on a scale that affects global chemistry. Think about what has to happen to suppress tree growth from California to Ireland to Chile, reduce solar radiation reaching Earth's surface for multiple consecutive years, temperature drops of several degrees, disrupted precipitation patterns, the kind of climate forcing that only comes from massive atmospheric loading with reflective particles, volcanic winter scenarios, nuclear winter models, large impact events. These are the mechanisms capable of global dimming. And think about what has to happen to crash a population by 99%. Disease epidemics can do it, but they don't show up in ice cores or tree rings. Warfare can reduce populations locally, but not across entire continents simultaneously. Famine can kill millions, but famine severe enough to reduce breeding populations to 500 individuals requires environmental collapse so complete that agriculture becomes impossible for years. The Maya recorded this. They called it the Great Flood. Every Maya creation text, the Popol Vuh, the Shalom Balaam books, the inscriptions at multiple sites, they all reference a catastrophic flood that destroyed the previous world. These weren't generic flood myths borrowing from biblical traditions. The texts were written based on oral traditions that predated Spanish contact, and the details are specific, not gradual rising waters, but sudden, violent deluge, not local flooding, but world-encompassing destruction, not metaphorical cleansing, but literal annihilation of populations. The Popol Vuh describes it explicitly. A great flood was made, which came upon the mannequins of wood. The face of the earth was darkened, and a black rain began to fall by day and by night. Black rain, that's not normal precipitation. That's ash-laden water, the kind that falls after massive atmospheric injection of particulates. The earth was darkened, and it began to rain night and day. Prolonged darkness, extended precipitation, climate disruption lasting months or years, not days. The text continues with details that sound less like mythology and more like eyewitness accounts transmitted through generations. They were destroyed by a resinous, thick rain. Resinous, sticky, tar-like precipitation. This description matches accounts from modern volcanic disasters where ash combines with rain to create dense, suffocating mudflows. Great and small animals, trees, rocks, everything was beaten and crushed and broken. Complete environmental devastation, ecosystems collapsing, the kind of wholesale destruction that paleontology recognizes from extinction events. Flood myths appear in cultures worldwide, and skeptics correctly note that independent invention of flood stories is common. Humans live near water. Rivers flood. Tsunamis happen. Societies develop stories explaining these recurring natural disasters. But the Maya flood narrative includes details that go beyond typical flood mythology. The darkness, the extended duration, the black rain, the global scope, the complete extinction of previous populations. These elements match the environmental signature from ice cores and tree rings. Other civilizations show similar disruption during the same time window. Egyptian records from the late New Kingdom describe years of darkness and famine around 1200 BCE. Chinese texts reference prolonged cold and failed harvests during the late Shang dynasty, roughly 1100 BCE. Hittite records document crop failures and population movements. The Bronze Age collapse, one of the most significant civilizational disruptions in ancient history, occurred between 1200 and 1150 BCE. Dozens of empires and city-states across the eastern Mediterranean collapsed within decades, not from conquest, but from environmental and social pressures that remain incompletely explained. The global pattern suggests a common trigger, something affecting climate worldwide, disrupting agriculture, forcing population movements, collapsing complex societies simultaneously across continents. The ice cores, tree rings, and genetic bottlenecks indicate this trigger occurred around 1150 BCE. The Maya flood myths describe experiencing it, and the timing correlates precisely with when Southeast Asian genetic markers begin appearing in the Maya gene pool, not before the catastrophe, after. The oldest Maya samples showing Southeast Asian DNA date to approximately 1000 BCE, within a century of the environmental crisis. Before that date, genetic samples show only expected Native American lineages. The trans-Pacific genetic markers appear suddenly in multiple individuals at multiple sites right after the population bottleneck. The founder effect analysis suggests these lineages arrived in a single migration event or series of closely timed events involving 40 to 100 individuals. Populations don't undertake trans-Pacific voyages carrying dozens of people without compelling reasons. Maritime technology capable of that scale of ocean crossing requires enormous resources, specialized knowledge, months or years of preparation. You don't accidentally assemble that capability. You don't casually decide to sail 8,000 miles into the unknown. Something has to drive populations to abandon their homelands and risk everything on journeys that most participants won't survive. Environmental catastrophe is that kind of driver. When the environment you've adapted to suddenly becomes uninhabitable, when crops fail for years, when the sky darkens and rain turns black, when populations crash by 99%, survivors don't stay put, hoping things improve. They move. They seek habitable zones. They undertake desperate voyages to anywhere that might support life. The archaeological record in Southeast Asia shows exactly this pattern. Settlement abandonments. Around 1100 BCE, coastal villages depopulated. Evidence of rapid migration inland and toward higher ground. Populations relocating from lowland river valleys to upland refuges, and in some cases, disappearing from the archaeological record entirely. Not dying in place, but leaving, going somewhere that didn't preserve evidence of their arrival because we weren't looking in the right places, until we looked in Mesoamerica and found their DNA. The settlement pattern in the Maya lowlands shows something that has puzzled archaeologists for decades. Before 1000 BCE, population density was low, small scattered villages practicing basic agriculture. Then suddenly, around 900 BCE, settlement density explodes. Population increases dramatically. Architectural complexity jumps from simple pole and thatch structures to stone platforms and ceremonial buildings. Pottery styles diversify. Trade networks expand. All the markers of civilizational acceleration appear in a compressed time frame. Standard interpretation attributes this to internal development. Agricultural intensification allowing population growth, social complexity emerging from increased population density, specialization and hierarchy developing as societies grow. But those processes usually take centuries, not decades, and they usually happen gradually, not as sudden transitions visible in the archaeological record. An alternative interpretation fits the data better. A catastrophe decimated existing populations. Survivors scattered. Then new populations arrived, bringing knowledge and technology from elsewhere, merging with surviving local groups, and rapidly building new settlements, using a combination of local and imported practices. This produces exactly the pattern we see: sudden population increase despite recent bottleneck, rapid architectural advancement, cultural practices that blend local traditions with foreign influences, genetic diversity that exceeds what truncated local populations could generate. The genetic data from Maya burials shows this fusion. Individuals with pure Native American ancestry buried alongside individuals with mixed ancestry, buried alongside rare individuals with predominantly Asian ancestry, not segregated, not a conquering population replacing an indigenous one, integrated, multiple genetic backgrounds sharing burial grounds, suggesting social integration, intermarriage, combined societies building something new from multiple sources. This wasn't conquest, it wasn't colonization, it was refuge and merger. Populations fleeing environmental collapse in Asia, arriving in Mesoamerica that was also depopulated by the same global catastrophe. Survivors from different regions encountering each other in the aftermath and choosing cooperation over conflict because both groups were desperate, combining knowledge, technology, and genetics to rebuild in a world where the old order had been destroyed. The Maya flood myths preserve this memory, not just the catastrophe, but what came after. The Popol Vuh describes how after the flood destroyed the mannequins of wood, new peoples came from the east and became the first fathers of Maya civilization. New arrivals after destruction from the east, which from a Mesoamerican perspective means across the Pacific, becoming founders of what followed. They weren't explorers seeking new territory. They were survivors fleeing from something that left scars in the ice, the trees, and the genetic code itself. Environmental disaster that triggered one of the great human migrations, largely unrecorded by history because it happened in an era before widespread literacy, preserved only in oral traditions that scholars dismissed as mythology and genetic evidence that we're only now learning to read. In 2022, computational linguists ran every known Maya dialect through machine learning algorithms designed to detect language mixing and borrowing. The results suggested that Proto-Mayan didn't evolve from a single ancestral language. It was constructed, deliberately, from at least 4 distinct linguistic families. Language evolution follows predictable patterns. When populations remain isolated, their languages diverge gradually. Vocabulary changes, grammar shifts, pronunciation drifts. But the underlying structure stays recognizable. Spanish, Italian, French, Portuguese descended from Latin through centuries of gradual divergence. You can trace the family tree backward, reconstruct the ancestral language, map how specific changes propagated through time. Mayan languages don't follow this pattern. The computational analysis used algorithms originally developed for detecting plagiarism and identifying machine translation artifacts. These tools excel at finding passages that don't belong, text that was copied from elsewhere rather than organically composed. When applied to linguistic databases, They can identify borrowed words, grammatical structures imported from other language families, phonological patterns that don't fit the expected evolution. When researchers at MIT and Universidad Nacional Autónoma de Mexico fed 31 Mayan languages and dialects into these systems, the algorithms flagged over 2,000 words as likely borrowings. Not recent loanwords from Spanish or English that obviously entered after European contact. Deep structural vocabulary, basic terms for kinship, body parts, natural phenomena, numbers. The kind of words that languages don't borrow because they already have them. The kind of words that should be the most stable and resistant to replacement. These flagged words didn't cluster into a single external source. The analysis identified loan words from at least 4 distinct language families: Proto-Austronesian from Southeast Asia and the Pacific, Tai-Kadai from southern China and mainland Southeast Asia, Niger-Congo from West Africa, and a 4th source that doesn't match any catalogued language family, but shows structural similarities to extinct Mediterranean languages. The statistical confidence on these identifications ranged from 78% to 94%. Those aren't perfect scores, but they're far above the noise threshold. When the same algorithms are applied to languages with known borrowing histories, like English taking words from Norman French, the detection accuracy is around 85%. The Mayan results fall in the same reliability range. Here's what makes this different from normal cultural mixing. All languages borrow words from neighbors. Spanish took from Arabic during Moorish rule. Japanese borrowed from Chinese. That's expected linguistic contact. But borrowed vocabulary usually comes in semantic clusters. Technical terms from cultures with advanced technology in that domain. Religious vocabulary from dominant faiths. Trade goods from economic partners. The borrowing is thematically coherent. The Mayan borrowings are distributed across all semantic categories. Kinship terms from one source, astronomical vocabulary from another, agricultural words from a third, basic verbs from a fourth. This isn't selective adoption of useful foreign concepts. This is wholesale linguistic fusion. Multiple language communities contributing core vocabulary to create a hybrid system that functioned as the lingua franca of a civilization. Grammar tells the same story. Mayan languages use ergative absolutive alignment, a grammatical structure where subjects of transitive verbs are marked differently from subjects of intransitive verbs. Only about 25% of world languages use ergative systems. Most use nominative, accusative alignment like English. Ergative alignment is common in Austronesian languages, Basque, some Caucasian languages. Its presence in Mayan fits the pattern of Austronesian contact suggested by the genetic data. But Mayan grammar also includes features typical of verb-initial languages, where the default word order places the verb before subject and object. This structure is common in Polynesian languages and some Celtic languages, and overlaying that are grammatical particles that function similarly to topic markers in East Asian languages like Japanese and Korean. The result is a grammatical system that blends features from multiple typologically distinct language families. Linguistic typology says this shouldn't happen. Languages typically belong to clear structural types. They might borrow vocabulary from other types, but core grammar stays relatively stable. Languages don't casually mix ergative alignment from one family with verb initial structure from another family with topic marking from a third. That kind of fundamental grammatical fusion requires sustained bilingualism or multilingualism over many generations, with speakers so fluent in multiple languages that they freely blend grammatical strategies. The only documented cases of comparable grammatical mixing come from Creole languages that emerged in colonial contact zones. Haitian Creole blending French vocabulary with West African grammar. Tok Pisin in Papua New Guinea mixing English with indigenous Melanesian structures. But Creoles form in specific socio-linguistic contexts. Populations forced into contact, power imbalances, creating need for communication. Simplified pidgin languages that children then nativize into full languages with more complex grammar. Did the Maya emerge from a similar contact scenario? Multiple populations arriving in the same region, needing to communicate, creating a hybrid language that their descendants elaborated into the sophisticated Mayan language family. The genetic clustering data supports this model. Admixture analysis, the computational method that identifies ancestral populations contributing to modern genomes, suggests Maya populations have genetic input from at least 7 distinct ancestral groups. When geneticists plot Maya genomes on principal component analyses, statistical visualizations that group genetically similar individuals, Maya samples don't cluster tightly. They spread across multiple clusters, suggesting population structure created by recent mixing, rather than long isolation. Each genetic cluster corresponds to different ancestry proportions. One cluster shows predominantly Native American ancestry with small Southeast Asian contribution. Another shows balanced Native American and East Asian ancestry. A third shows Native American baseline with West African and European contributions. And these aren't just individual outliers. They're coherent population subgroups, each representing a different mixture of the same source populations in varying proportions. This genetic structure indicates prolonged admixture over many generations, not a single contact event, but sustained interaction between multiple groups, some individuals having more ancestry from one source, some from another, but all groups interbreeding enough to create continuous genetic variation rather than distinct separated populations. The cultural archaeology shows the same pattern. Early pre-classic Maya sites show ceramic traditions that don't derive from a single ancestral pottery style. Instead, vessel forms, decoration techniques, and clay preparation methods show influences from multiple regions. Some ceramics resemble Polynesian pottery forms. Others use tempering techniques common in Southeast Asian ceramics. Some decoration styles match West African textile patterns, translated into clay. Architectural traditions show similar diversity. Maya corbelled arch construction appears suddenly in the archaeological record without local antecedents, but corbelled arches appear in Bronze Age Mediterranean architecture, in Mycenaean tholos tombs, in Etruscan buildings. The technique isn't difficult to invent independently, but the Maya versions include specific design elements that match Mediterranean examples more closely than expected from convergent evolution. Stone carving traditions show evidence of multiple stylistic sources. Some iconographic elements match Polynesian and Indonesian motifs. The way Maya sculptors depicted water and waves resembles artistic conventions from Southeast Asian Buddhist art. Representations of supernatural beings combine attributes that appear in mythologies from different continents. This isn't vague similarity that could result from universal human symbolism. These are specific artistic conventions, technical approaches stylistic choices that match source populations where the genetic markers originated. The correlation between genetic ancestry, linguistic borrowing, and cultural practice is consistent. All three lines of evidence point to multiple populations contributing to Maya civilization. The timeline of arrivals matters. The genetic data suggests multiple waves. The earliest Southeast Asian markers appear around 1000 BCE, right after the environmental catastrophe. African genetic markers appear slightly later, around 800 BCE. European Y chromosome lineages show up around 600 BCE. Each wave small in absolute numbers, but large enough to contribute lasting genetic signatures. Were these separate, independent arrival events, or coordinated migrations of multi-ethnic groups that had already mixed in other locations before reaching Mesoamerica? The answer isn't clear, but the pattern of admixture suggests populations were already interacting before arrival. The genetic mixing isn't happening sequentially in Mesoamerica. It's appearing already mixed, as if the groups that arrived had already formed multi-ethnic communities elsewhere, then relocated those communities to the Maya region. That scenario fits the linguistic evidence better. If Proto-Mayan was created from scratch in Mesoamerica by populations that arrived speaking distinct languages, we'd expect to see more evidence of the pre-contact languages persisting. Substrate languages influencing Maya, but remaining distinct. Code switching between languages. Bilingual communities maintaining both systems. Instead, we see a single elaborated language family that appears fully formed with mixed structural features already integrated. The social organization of classic Maya cities reveals stratification that correlates with genetic ancestry. Elite burials show higher frequencies of non-Native American genetic markers. Commoner burials show predominantly Native American ancestry. This isn't absolute segregation. There's overlap, intermarriage, social mobility. But the statistical pattern is clear. The ruling class concentrated foreign genetic ancestry, while commoner populations remain more demographically stable. But here's what that pattern doesn't show. It doesn't show conquest by foreign populations subjugating indigenous peoples. Conquest produces different genetic signatures. Y chromosome lineages from conquerors flooding into local populations, while mitochondrial DNA remains indigenous, indicating foreign men taking local women. The Maya pattern shows both male and female lineages from multiple sources integrating at multiple social levels. Not conquest, but coalition. Different groups coming together, contributing different types of knowledge and skills, organizing into hierarchies based on specialized expertise rather than pure military dominance. The specialized knowledge each group contributed is documented in the archaeological record. Astronomical knowledge reaching levels that suggest input from multiple observation traditions. Mathematical concepts, including place value notation, and zero appearing without local developmental sequence. Agricultural techniques adapted to multiple crop complexes from different geographic origins. Maritime technology beyond what riverine populations would develop. Metallurgical knowledge for working jade and other hard stones, using techniques that match Asian workshops. Medical practices including surgical procedures documented in skeletal remains that match techniques from distant regions. These aren't a few borrowed ideas. This is comprehensive knowledge transfer across multiple domains, the kind of knowledge integration that requires sustained teaching, apprenticeship, translation not just of words, but of entire conceptual frameworks. It requires populations remaining in contact for generations, not brief encounters or isolated traders. The later classic period texts contain explicit references to distinct lineages, maintaining separate identities while functioning within unified political structures. Inscriptions name ruling families by their origins. The people who came from across the great water, the lineage of the eastern homeland, the descendants of the 7 caves founders. These aren't metaphorical ancestral references. They're contemporary social identifiers used by populations that remembered and maintained distinct origin narratives even after centuries of intermarriage and integration. The purpose of this multi-ethnic fusion is suggested by the timing and achievements. The most sophisticated Maya developments, the advanced astronomy, the complex mathematics, the monumental architecture, the hieroglyphic writing, all emerge after the genetic and linguistic mixing is documented. Before the fusion, during the early pre-classic period, Maya sites show modest villages with limited architectural complexity and no evidence of literacy or advanced astronomy. After the fusion, within a few centuries, the classic Maya civilization reaches heights that rival and, in some ways, exceed contemporary Old World civilizations. The implication is uncomfortable, but clear. Maya civilization, as we know it, wasn't built by a single indigenous population developing in isolation. It was built by deliberate fusion of multiple populations bringing knowledge from multiple source regions, combining that knowledge into something greater than any single tradition could have produced alone. A planned synthesis, creating a civilization designed to preserve and advance knowledge from multiple global sources. They weren't random refugees fleeing disaster. They were selected populations, chosen for specific knowledge and skills, brought together with purpose, building not just a new society, but an archive, a library of human knowledge encoded in genetics, language, architecture, and astronomical observations, preserved in a remote location far from whatever catastrophe had destroyed the world they came from. And if that's true, if Maya's civilization was a deliberate knowledge preservation project involving populations from across the planet, then the mass graves make horrifying sense. Not random violence, not political purges, but population correction, eliminating genetic lines that didn't fit the plan, concentrating traits deemed valuable, removing those deemed incompatible. Genetic engineering at civilizational scale, running for centuries, producing the bloodlines that would rule one of the most sophisticated societies in the ancient world. Let me take you deeper into the evidence that is keeping bioethicists awake at night. Not the speculation, not the theories, but the actual measurements that do not make sense, no matter how many times you run the numbers. Start with the sacrifice victims. Maya civilization practice human sacrifice. This isn't controversial. Stone carvings depict it. Archaeological remains confirm it. Skeletal trauma patterns show it. But what archaeologists assumed was random ritual killing, selecting victims based on availability or social status or religious calendar needs, turns out to have been something far more systematic when you analyze the genetic data. A 2023 study published in Nature Human Behavior examined DNA from 156 individuals identified as probable sacrifice victims based on skeletal trauma patterns and burial context. The victims came from 12 different Maya sites spanning 600 years, from 200 AD to 800 AD, standard archaeological analysis focused on age, sex, skeletal pathology, trauma patterns, the usual bioarchaeological variables. But when genetic analysis was added, patterns emerged that conventional methods couldn't detect. The sacrifice victims showed significantly higher genetic diversity than contemporary elite burials at the same sites. Average heterozygosity in victim populations was 0.64 compared to 0.42 in elite lineages. Remember what that means. Higher heterozygosity indicates more genetic variation, less inbreeding, more diverse ancestry. The people being sacrificed weren't the inbred nobility. They were genetically diverse individuals from outside the elite breeding networks. More specifically, the victims showed haplogroup distributions that deviated systematically from both elite and commoner population baselines. They carried rare genetic variants at frequencies that shouldn't occur by chance. When researchers calculated the expected frequency of specific alleles based on overall population genetics, then compared that to observed frequencies in sacrifice victims, the differences reached statistical significance at p-values below 0.001. In plain language, the genetic profiles of victims were so unusual that random selection from the general population couldn't explain the pattern. Less than 0.1% chance this happened randomly. The age and sex distributions add another layer. Of the 156 victims analyzed, 68% were between ages 15 and 25 at death. Only 8% were children under 10. Just 4% were over 40. Both sexes represented roughly equally, 52% male, 48% female. This isn't a natural mortality distribution. Disease and famine kill the very young and very old preferentially. Warfare kills young males. This demographic profile suggests selection criteria. Prime reproductive age individuals of both sexes. Now layer in the skeletal markers. The victims showed specific physical characteristics at frequencies that exceeded population baselines. Cranial measurements clustered in ranges that anthropologists associate with particular ancestry components. Dental trait combinations indicated genetic backgrounds. Bone density measurements, skeletal robusticity, limb proportions, all showed patterns suggesting victims were selected partly based on physical appearance that correlates with underlying genetics. Here's what makes this discovery so powerful. The Maya had no microscopes, no DNA sequencing, no laboratory techniques for detecting invisible genetic markers, but they could see physical traits. They could observe that certain families consistently produced children with particular facial structures, body proportions, dental patterns. They could track these traits through lineages and identify individuals carrying them. And apparently, they systematically eliminated those individuals. The genetic variants being removed weren't random. Statistical analysis identified specific genomic regions that showed depleted diversity in post-sacrifice population samples compared to pre-sacrifice samples from the same sites. These regions include chromosome 15 around the OCA2 and HERC2 genes affecting pigmentation, chromosome 7 near the FGFR2 gene involved in cranial development, and chromosome 4 around the SNCA gene associated with neural development. Pause and think about what this suggests. The Maya were identifying individuals carrying specific genetic variants that affected observable physical and possibly behavioral traits, then removing those individuals from the breeding population through ritual sacrifice. Not indiscriminate killing. Genetic selection masked as religious practice. The scale of this program is documented in burial records. Conservative estimates based on excavated sacrifice sites suggest 50,000 to 100,000 individuals were ritually killed during the Classic period. That's not total Maya sacrifices, which were certainly higher. That's the subset showing the specific demographic and genetic profiles indicating systematic selection. 50,000 people killed based on criteria that included their genetic makeup. Comparison to modern genetic selection reveals uncomfortable parallels. Contemporary genetic engineering focuses on identifying desirable alleles and increasing their frequency while reducing undesirable variants. CRISPR, gene editing, embryo selection in IVF, genetic screening programs. These are modern tools, but the underlying logic is identical. Identify genetic variants affecting traits of interest. Increase frequency of desirable variants. Decrease frequency of undesirable variants. The Maya were doing this without molecular biology, using observable physical traits as proxies for underlying genetics and modifying population genetics through elimination. The geographic variation in this practice tells us something important. Not all Maya sites show the same patterns. Tikal, Copan, Calakmul, the major power centers, these sites show strong genetic selection signatures. Smaller sites, peripheral cities, village settlements, they show much less evidence of systematic sacrifice based on genetic criteria. The practice was concentrated in political and religious centers among populations with enough organizational capacity to maintain multi-generational selection programs. The timeline of intensification tracks the rise of classic Maya civilization. Before 200 AD, evidence of genetic selection through sacrifice is minimal. Between 200 and 600 AD, the practice intensifies gradually. From 600 to 800 AD, at the height of classic Maya power, the genetic selection reaches its peak. Sacrifice rates increase. Genetic profiles of victims show stronger deviation from population norms. The demographic patterns become more precise. Then, after 800 AD, during the terminal classic collapse, the practice breaks down. Sacrifice continues, but the genetic selectivity disappears. The system that maintained multi-century population engineering fell apart, along with classic Maya civilization. The correlation between genetic engineering intensity and civilizational achievement is stark. The centuries when sacrifice-based genetic selection was most systematic are exactly the centuries when Maya astronomy, mathematics, architecture, and writing reached their zenith. The sites showing the strongest genetic selection signatures are the sites with the most sophisticated astronomical observations, the most complex mathematical inscriptions, the most elaborate architectural achievements. Does this prove genetic selection caused Maya achievements? No. Correlation isn't causation. But it's difficult to ignore the relationship. For 600 years, Maya civilization systematically concentrated specific genetic variants in elite lineages, while eliminating other variants from the population through sacrifice. During those same 600 years, Maya intellectual and technological accomplishments exceeded anything in pre-Columbian Americas and rivaled contemporary Old World civilizations. When the genetic selection program collapsed, classic Maya civilization collapsed with it. Physical evidence from sacrificial contexts shows the ritual wasn't random violence disguised as religion. The architectural settings where sacrifices occurred were designed with astronomical alignments, specific dates in the Maya calendar correlating with celestial events, chamber orientations that created visual effects during solstices or equinoxes. The level of planning and coordination suggests these weren't spontaneous religious expressions. They were scheduled, organized, systematized programs running continuously across multiple sites for centuries. Skeletal analysis reveals another disturbing detail. Many sacrifice victims show evidence of extended captivity before death. Skeletal stress markers indicating dietary restriction, bone changes from prolonged immobilization, dental pathology suggesting altered diet composition. These individuals weren't captured and immediately killed. They were held, possibly evaluated, certainly observed, before being selected for sacrifice. The process included assessment periods, where decisions were made about who would be eliminated based on criteria that extended beyond immediate physical appearance. Modern discovery of these patterns happened recently because the analytical tools only became available in the last decade. Ancient DNA extraction techniques that can recover genetic material from degraded remains, computational methods that can detect subtle statistical patterns in large datasets, population genetic models sophisticated enough to identify selection signatures, bioarchaeological protocols that integrate genetic, skeletal, and contextual data. These capabilities didn't exist 20 years ago. The evidence was always there in the ground. We just couldn't read it. Scientific reluctance to publish implications is documented in the publication history. The genetic data showing systematic selection signatures first appeared in supplementary materials of papers focused on other topics, population structure studies that mentioned unusual victim demographics in footnotes, bioarchaeological reports that noted genetic deviations but didn't interpret them. Researchers collected the data, saw the patterns, then carefully avoided stating the obvious conclusion that Maya civilization practiced genetic engineering through elimination at scales that wouldn't be matched until 20th century totalitarian regimes. When the Nature Human Behavior paper finally addressed the implications directly, the response from the broader archaeological community was muted. Few citing papers, limited media coverage, no follow-up studies announced. The findings didn't spark the intense debate you'd expect from a discovery this significant. The silence suggests institutional discomfort with acknowledging that one of archaeology's celebrated civilizations was built partly on systematic genetic selection through ritual murder. The ethical questions this raises for how we interpret ancient civilizations are profound. We celebrate Maya achievements in astronomy and mathematics. We study their art and architecture as pinnacles of pre-Columbian culture. Museum exhibitions present Maya civilization as sophisticated and admirable, all of which is true. But those achievements were accomplished by a society practicing genetic engineering through mass sacrifice, by elite classes that concentrated foreign genetic ancestry while eliminating indigenous genetic variants, by political structures that maintained population control programs for centuries. Modern genetic engineering raises parallel ethical concerns. Should we edit human embryos to eliminate disease genes? Should we screen for cognitive enhancement alleles? Should we allow parental choice in offspring genetics? These debates assume genetic modification is a future possibility requiring careful consideration. But the Maya already did it. They demonstrated that human populations can be genetically modified without molecular biology, using nothing more than systematic selection over generations. And they showed that societies willing to kill tens of thousands to achieve genetic goals, can produce remarkable civilizations. That's the uncomfortable truth. Genetic engineering and cultural achievement aren't incompatible. They might even be linked in ways we don't want to acknowledge. If you're willing to eliminate genetic variants through violence, if you're willing to concentrate traits through controlled breeding, if you're willing to maintain these programs for centuries, you can reshape human populations. And those reshaped populations might achieve things that naturally evolved populations don't. The question isn't whether genetic selection through elimination works. The Maya proved it works. The question is whether we're comfortable admitting that one of history's great civilizations was built on a foundation of systematic genetic violence. Whether we can hold both truths simultaneously, that Maya civilization was brilliant and beautiful, and that it was also eugenic and brutal. Right now, dozens of Maya sites have genetic samples waiting to be analyzed, burial chambers not yet opened, DNA extraction techniques improving every year. Each excavation season brings new remains into laboratories. By 2030, we might have genetic data from 5,000 Maya individuals spanning 2,000 years. That dataset will reveal exactly what the genetic selection program was doing, which traits were being selected for and against, how successful the program was at reshaping population genetics, whether the traits being concentrated actually correlated with the capabilities that made Maya civilization distinctive. The December 2025 excavation at Tikal is expected to uncover additional sacrifice contexts. New DNA analysis, results from Copan, scheduled for March 2026, will include samples from previously unexamined tomb complexes. And somewhere in the Guatemalan jungle, there's a sealed chamber that ground-penetrating radar has identified, but excavators haven't reached yet. A chamber that Maya builders deliberately concealed, filled with remains, then buried under layers of construction that suggest they wanted whatever is inside to stay hidden until someone with the technological capability to understand it could dig it up. That someone is us. That technology is now. And what we're discovering is darker than we imagined. Not a civilization that rose through normal development and fell through normal collapse, but a deliberately engineered society that assembled populations from across the planet, fused their genetics and knowledge through planned interbreeding, concentrated desired traits through systematic selection, and eliminated undesired variants through ritualized violence spanning centuries. An experiment in human genetic and cultural synthesis operating at civilizational scale. The Maya told us where they came from. They told us they were assembled from multiple distant origins. They recorded their breeding lineages and origin stories in texts we dismissed as mythology. They built monuments documenting their astronomical knowledge and mathematical sophistication. They left physical evidence of exactly what they were doing in mass graves, in genetic signatures, in skeletal markers of selection. We just weren't ready to believe that an ancient civilization could understand heredity well enough to practice genetic engineering, that they could organize systematically enough to maintain selection programs across centuries, that they could be both brilliant and brutal, sophisticated and savage, achieving remarkable intellectual heights while building on foundations of genetic violence. But the DNA doesn't lie. The bones don't lie. The statistical patterns don't lie. Maya civilization practiced genetic selection through elimination. They concentrated foreign ancestry in elite bloodlines while removing indigenous variants from the population. They killed based on genetic criteria they couldn't sequence, but could observe through physical traits passed through families. And in the process, they built one of the most sophisticated civilizations in human history, which raises a question that every future genetic engineering debate will have to grapple with. If genetic modification of human populations can produce civilizational achievements, if systematic selection can create capabilities that natural evolution doesn't, if societies are willing to do what the Maya did, what should stop them? Ethics, law, the knowledge that previous attempts ended in collapse? The Maya answer is written in their ruins. Their genetic engineering program ran for 600 years. Their civilization reached extraordinary heights. And then it ended. Cities abandoned, populations scattered, the carefully engineered bloodlines disrupted. Whether the collapse happened because of the genetic program or despite it, we don't yet know. But the trajectory is clear. Build a civilization on genetic selection, achieve remarkable things, then watch it fall apart when the selection system breaks down. Maybe that's the lesson. Not that genetic engineering doesn't work, but that civilizations built on it are inherently unstable. Their populations can tolerate systematic genetic violence for centuries, but eventually something breaks, social cohesion fractures, environmental pressures overwhelm engineered advantages. The very traits selected for become maladaptive when conditions change. And when collapse comes, it comes completely. The Maya left us evidence, physical, genetic, cultural, everything we need to understand what they did and what happened as a result. We're just beginning to read that evidence with tools sophisticated enough to reveal its full implications. And what we're finding is a mirror. A civilization that asked the same questions we're asking about genetic modification, answered them differently than our ethics currently allow, and left a 1,000-year-old experiment's results buried in the jungle waiting for us to excavate. Whether we learn from their example or repeat their mistakes remains to be seen. Right now, as you read this, laboratory technicians at 3 separate ancient DNA facilities are processing samples that could rewrite everything we thought we knew about human civilization. Not Maya samples, not even American samples. These are remains from Egypt, Mesopotamia, the Indus Valley, Shang Dynasty China. Sites separated by thousands of miles, cultures that supposedly developed in isolation. And preliminary genetic results are showing the same impossible signatures that appeared in Maya DNA, transoceanic haplogroups, multi-continental ancestry, evidence of deliberate population mixing during the exact same time windows. The pattern isn't unique to Mesoamerica, it's global. In November 2024, a research team analyzing DNA from early Egyptian dynastic burials detected haplogroups from Southeast Asia and Sub-Saharan Africa appearing simultaneously around 3100 BCE, the first dynasty when Egypt unified and writing emerged, when monumental architecture suddenly appeared, when sophisticated administrative systems materialized seemingly overnight. The genetic data suggests this wasn't internal development. It was population fusion, multiple groups combining knowledge and genetics to create something unprecedented. December 2024 brought results from Mohenjo-Daro, one of the great Indus Valley cities. Ancient DNA from elite burials dated to 2600 BCE showed ancestry components from the Near East, Central Asia, and unexpectedly East Africa and Southeast Asia. The same multi-continental genetic mixing that produced Maya civilization was happening in South Asia at the same time. Different continents, different cultures, same process. January 2025 added Shang Dynasty oracle bone inscriptions. New linguistic analysis using the same computational methods applied to Mayan languages revealed loan words from Proto-Indo-European, Semitic languages, and Austronesian sources. Bronze Age China incorporating vocabulary from populations that shouldn't have been in contact. The oracle bones themselves, used for divination and recording, show mathematical notation systems with structural parallels to both Maya and Egyptian number systems. Not identical, but related, as if all three derive from common source material. The picture emerging from genetic, linguistic, and archaeological data across multiple civilizations points towards something that shouldn't be possible under current historical models. During a narrow window between 3000 and 1500 BCE, multiple sophisticated civilizations emerged on different continents, all showing evidence of population mixing from distant sources, all developing similar knowledge systems, all building monumental architecture using comparable techniques, all creating writing systems within centuries of each other. Either this is the most extraordinary coincidence in human history, with independent populations across the planet simultaneously developing identical solutions to identical problems using identical population assembly strategies. Or something else was happening. Something coordinated. Something that involved moving populations, sharing knowledge, and deliberately engineering societies across the ancient world. The Maya weren't an isolated experiment. They were part of a global pattern. Think about what that implies. Not random migrations driven by environmental pressures, not gradual cultural diffusion through trade and contact, but organized, deliberate assembly of civilizations, using populations selected from across the planet. For what purpose? To preserve knowledge after the catastrophes that show up in ice cores and tree rings, to create redundant archives of human achievement distributed globally in case any single region faced destruction, to conduct massive social experiments testing different combinations of genetic and cultural elements. We don't know, but we're about to find out more. The December 2025 excavation at Tikal isn't just uncovering more Maya remains. The team is specifically targeting a structure that ground-penetrating radar suggests contains a sealed chamber beneath the main temple complex. The chamber's size and depth indicate it wasn't a typical burial. It's too large, too carefully concealed, too deliberately positioned at the architectural center of the city. Initial probing suggests the chamber contains not just human remains, but artifacts, possibly written records, definitely astronomical instruments based on metal detection patterns. If the chamber contains what archaeologists suspect, if Maya builders deliberately left a time capsule documenting their knowledge and origins, if the preservation conditions are good enough that organic material survived, we can have Maya texts describing exactly what they were doing, who organized it, and what they knew about the global pattern of civilization building. The excavation is scheduled for December 2025, 18 months from now. The team includes geneticists, linguists, archaeoastronomers, materials scientists. They are planning to live stream the opening. Whatever is inside that chamber, the world will see it simultaneously. No institutional filtering, no carefully worded academic papers published years later. immediate documentation of what 1,200 years of sealed darkness preserved. March 2026 brings the Copan results. DNA analysis from 200 newly excavated individuals spanning the entire Classic period. These aren't random burials. The excavation targeted specific residential compounds associated with Maya astronomers and scribes based on inscription analysis. The individuals buried there weren't warriors or kings. They were the intellectuals, the mathematicians and calendar keepers and writers who produced the codices and almanacs and astronomical tables. Their DNA will reveal whether the knowledge class showed different genetic profiles than military or political elites, whether cognitive abilities were being selected for as intensely as physical traits, whether the breeding program focused on creating intellectual dynasties capable of maintaining and advancing the scientific traditions. The implications extend beyond archaeology and genetics into contemporary debates about human enhancement. If Maya genetic selection actually worked, if concentrating specific alleles through controlled breeding and elimination produced populations with enhanced capabilities in astronomy, mathematics, or other cognitive domains, that changes the ethical calculus around modern genetic modification. It means genetic engineering of human cognitive abilities isn't theoretical. It's been done. We're just rediscovering the results 1,000 years later, and if it worked for the Maya, if systematic selection over 20 to 30 generations can measurably enhance cognitive capabilities, then every argument against human genetic enhancement has to grapple with historical precedent. The Maya demonstrate that genetic modification through selection is possible, that it can produce civilizational benefits, and that societies are willing to pay the cost in systematic violence if the perceived rewards are high enough. Do we want to repeat that experiment with modern tools? CRISPR gene editing is faster than selective breeding. Embryo selection is cleaner than ritual sacrifice. But the underlying logic is identical. Identify desirable genetic variants, increase their frequency, reduce undesirable variants. The methods differ. The goal remains the same: creating human populations optimized for specific capabilities. The Maya give us a case study in where that leads. 600 years of increasing sophistication, remarkable achievements in science and mathematics, monumental architecture that still stands, and then collapse, cities abandoned, populations dispersed, the carefully engineered bloodlines disrupted. Whether genetic selection caused the collapse or merely failed to prevent it, the endpoint is the same. Civilizations built on genetic engineering prove unstable over long timescales. Maybe the problem is that selecting for specific traits creates populations adapted to specific conditions. When those conditions change, when environmental pressures shift, when new diseases emerge, when climate fluctuates, the genetically homogeneous elite lack the diversity to adapt. Their carefully concentrated traits become liabilities. The commoner populations with greater genetic diversity survive and disperse. The engineered elite die out. The civilization collapses because the ruling class that maintained it no longer exists. Or maybe the problem is social. Populations tolerate systematic genetic violence during good times when the benefits are visible. Advanced astronomy provides better agriculture through precise calendar predictions. Mathematical sophistication enables more efficient resource management. Monumental architecture demonstrates power and attracts trade. But when environmental stress hits, when drought reduces food production, when trade networks collapse, when the benefits of the genetic program become less apparent, while the costs in human sacrifice remain constant, social cohesion fractures. People stop accepting that sacrificing their relatives serves the greater good. Resistance builds. The system breaks down. The skeletal evidence from terminal classic sites shows both processes occurring simultaneously. Elite populations declining due to environmental stress and disease. Evidence of violent conflict increasing. Sacrifice rates initially accelerating as elites try to maintain control, then abruptly ceasing as the institutional structures necessary for organized killing collapse. The genetic engineering program didn't gradually wind down. It crashed, hard and fast, taking classic Maya civilization with it. Contemporary genetic engineering projects should study that trajectory. Not just the 600 years of success, the sudden failure, the inability to adapt when conditions changed, the social breakdown when costs exceeded perceived benefits, the complete system collapse rather than gradual transition. These are the risks that historical precedent documents. But there's another possibility that the Maya evidence suggests, one that's even more uncomfortable. What if the collapse was intentional? What if the civilization was always meant to be temporary? A knowledge preservation project designed to last until certain conditions were met, then deliberately shut down. The timing of the collapse correlates with astronomical cycles that Maya calculations tracked obsessively. The end of a major calendar round, specific planetary alignments, the kind of celestial events that Maya texts describe as significant transition points. If the civilization was built to preserve knowledge until a specific date, if the genetic engineering program was designed to produce populations capable of maintaining that knowledge for a specific duration, then the collapse at the end of that period makes sense. Not failure, but completion. Mission accomplished, knowledge preserved and encoded in monuments, texts, and genetics for whoever came next. We might be who came next. The civilization with the technological capability to read what they left, to sequence ancient DNA and decode genetic programs, to analyze astronomical alignments and recognize sophisticated mathematical systems, to excavate sealed chambers and read preserved texts. Everything the Maya did might have been designed for us, for this moment, when humanity has the tools to understand what they were and what they knew. The sealed chamber at Tikal might contain the message, the documentation explaining what Maya civilization was, who built it, what knowledge they were preserving, and what they expected us to do with it. If you were encoding information for a future civilization, you'd put it in the most obvious place, the largest city, the most prominent temple, sealed and protected but ultimately findable by anyone sophisticated enough to look. And you'd encode the same information in multiple formats, in written texts for literate civilizations, in astronomical alignments for scientifically advanced cultures, in genetic markers for societies with molecular biology, in monumental architecture for anyone capable of recognizing mathematical relationships, multiple redundant encoding systems ensuring the message survives even if some channels fail. The Maya left all of these. Texts were still deciphering. Astronomical observations were still analyzing. Genetic signatures we've only recently detected. Mathematical relationships in architecture we're only beginning to recognize. Every year we extract more information from evidence that's been sitting in plain sight for 1,000 years. Each analytical advance reveals another layer of encoding, another message waiting to be read. What happens when we finish reading? When we've decoded all the messages, sequenced all the DNA, analyzed all the inscriptions, excavated all the chambers? When we understand exactly what the Maya were doing, why they were doing it, and what knowledge they were preserving, do we use that knowledge? Do we attempt to recreate what they built? Do we implement our own genetic engineering programs using modern tools and Maya blueprints? Do we try to pick up where they left off, continuing the project they started 3,000 years ago? Or do we recognize their collapse as a warning, evidence that genetic engineering at civilizational scale, no matter how sophisticated, eventually fails? That the costs in systematic violence and social instability always exceed the benefits? That creating genetically optimized populations produces short-term gains but long-term catastrophe? The choice isn't theoretical. We have the technology now. CRISPR gene editing works. Embryo genetic screening works. If we wanted to implement a program to concentrate specific cognitive or physical traits in human populations, we could. Not through sacrifice, like the Maya. Through selection of embryos, through editing of germlines, through reproductive technologies that achieve the same ends with less visible violence. China is already experimenting with genetic enhancement. The 2018 announcement of CRISPR-edited babies proved that technical and ethical barriers to germline modification are lower than most assumed. If one country develops genetically enhanced populations with measurably superior cognitive or physical capabilities, other countries will face pressure to compete. The genetic engineering arms race could begin within decades, and the Maya evidence provides both encouragement and warning. Yes, genetic selection can work. Yes, enhanced populations can achieve remarkable things. And yes, civilizations built on genetic engineering eventually collapse. The question isn't whether we'll find more evidence of what the Maya did. We will. Every excavation season brings new data. Every genetic analysis reveals more patterns. Every computational linguistic study decodes more borrowed words. The evidence accumulates faster than we can publish it. By 2030, we'll have comprehensive documentation of Maya population genetics, complete linguistic reconstruction of their language sources, full architectural analysis of their astronomical alignments. We'll know exactly what they built and how they built it. The question is whether we're ready for what that evidence will reveal about how advanced civilizations actually emerge, what they're willing to do to maintain themselves, and what happens when genetic knowledge falls into hands that see people as breeding stock rather than individuals with inherent worth. The Maya saw people as breeding stock. The genetic data proves it. The mass graves document it. The systematic selection spanning centuries demonstrates it. And they built one of the most sophisticated civilizations in human history, which means seeing people as breeding stock and building advanced civilizations aren't mutually exclusive. They might even be connected. That's the dark truth waiting in the Tikal chamber. Not just knowledge about astronomy or mathematics. Knowledge about what humans are willing to do to each other in pursuit of civilizational greatness. Evidence that our ancestors already conducted the experiments we're debating conducting. Proof that genetic engineering works, but comes with costs we might not want to pay. December 2025. The chamber opens. The world watches. And somewhere in the darkness beneath a temple built 1,200 years ago by genetically engineered populations assembled from across the planet, there's a message. Instructions, maybe. Warning, possibly. Or just documentation. A time capsule left by a civilization that knew it was temporary, that encoded its knowledge in multiple redundant formats, that deliberately preserved evidence for whoever came next with the technology to understand. That's us. We're next. We have the technology. And whatever the Maya left is waiting. The only question now is whether we'll have the wisdom to use what we find, or whether we'll repeat their mistakes and build our own genetically engineered civilization that lasts 600 years before collapsing into ruins for some future culture to excavate and wonder what we were thinking. As of January 2025, DNA samples from 847 Maya individuals sit in freezers at ancient DNA laboratories across 5 continents. Each sample represents years of excavation work, months of extraction protocols, weeks of sequencing preparation. The data they contain could answer every question this script has raised, or it could reveal patterns we haven't yet imagined asking about. The samples exist. The technology exists. The analysis is happening right now while you're listening to this. But the publication timeline for genetic studies runs 18 to 24 months, from sequencing to peer review to publication, which means discoveries made in laboratory analysis rooms in 2024 won't appear in scientific journals until 2026. The researchers analyzing Maya DNA today already know things the public won't learn for another year. They've already seen patterns in the data that will rewrite textbooks. They're already debating how to present findings that challenge fundamental assumptions about human history. What do they know that we don't? What are they seeing in those genetic sequences that makes them pause before publishing? What conversations are happening in laboratory meetings about how to frame discoveries that sound impossible even to scientists who made them? We won't know until the papers appear, but we can read the signals. Conference presentations where researchers show preliminary data, then carefully hedge their interpretations. Grant applications requesting funding to explore unexpected ancestry patterns in pre-Columbian populations. Job postings for postdoctoral positions specializing in complex admixture scenarios in ancient American genetics. The institutional infrastructure is being built to analyze and interpret data that doesn't fit existing models. That preparation suggests the data is even more extreme than what's already been published. The December 2025 Tikal excavation isn't just another archaeological project. It's being funded at levels typically reserved for major oceanographic expeditions or particle physics experiments. $15 million over 3 years. For context, a typical Maya archaeology project runs on $200,000 annually. The funding comes from sources that don't usually bankroll Central American archaeology. Defense agencies interested in understanding how ancient civilizations organize complex systems. Technology companies curious about knowledge preservation across generations. Foundations focused on human enhancement ethics, wanting case studies of genetic selection programs. Why are these entities investing in Maya archaeology? Because they've seen the preliminary data. They know what's likely to be found in that sealed chamber. And they understand that whatever is down there has implications far beyond academic interest in pre-Columbian civilizations. This is applied research, studying Maya genetic engineering to inform contemporary debates about human enhancement, analyzing their knowledge preservation systems to design long-term information archives, understanding how they organized multi-generational selection programs to evaluate whether similar programs could be implemented today. The excavation team includes geneticists, but also bioethicists, population modelers, and specialists in genetic engineering regulation. That's not a typical archaeological crew. That's the team you assemble when you expect to find evidence that will immediately impact policy debates about human genetic modification. When you need people who can translate ancient data into contemporary implications, when you are preparing to present findings to audiences beyond academia. The chamber's contents, whatever they are, will be analyzed in real time and streamed to partner institutions globally, not for public transparency, for immediate expert analysis by teams positioned to interpret findings as they emerge. The public live stream shows the excavation. The private data feeds go to laboratories where genetic sequences can be analyzed, inscriptions translated, astronomical calculations verified, all within hours of discovery rather than years of careful study. That's unprecedented. Archaeological data traditionally goes through years of analysis before publication. The Tikal chamber contents will be interpreted on a timescale measured in weeks. Someone wants answers fast. Someone is willing to fund rapid analysis because time matters, because decisions need to be made soon, because whatever the Maya did, whatever knowledge they preserved, whatever warnings or instructions they left, multiple powerful institutions believe it's immediately relevant. What do they expect to find that's so urgent? The funding documents hint at possibilities without stating them explicitly. References to verification of historical genetic engineering protocols, mentions of ancient knowledge, preservation for long-duration crisis scenarios, language about precedent cases for organized human enhancement programs. These aren't phrases you use for routine archaeology. These are phrases indicating someone believes the Maya left a playbook, instructions for conducting genetic selection at civilizational scale, documentation of what worked, what failed, what costs they paid, what benefits they achieved. And multiple institutions want that playbook, want to know if genetic enhancement actually improved cognitive capabilities, want to understand the logistics of maintaining selection programs across centuries, want to learn what caused the collapse so they can avoid those failure modes. The Maya aren't just a historical curiosity. They're a case study, an experiment that ran for 600 years, documented its methods, preserved its data, and left the results for whoever came next to analyze and potentially replicate. The March 2026 Copan results will reveal whether the knowledge class showed different genetic profiles than other elites, whether the Maya were selecting specifically for cognitive traits, or whether intellectual achievement emerged as a side effect of selecting for other characteristics. That distinction matters enormously for contemporary genetic engineering debates. If cognitive enhancement came from selecting for physical traits that happen to correlate with intelligence, that's one scenario. If they were actively identifying and breeding for cognitive capabilities directly, that's something else entirely. One suggests genetic enhancement is feasible, but requires understanding complex trait interactions. The other suggests ancient civilizations had knowledge about heredity of cognitive abilities that we're only recently rediscovering. The Copan samples include individuals whose inscriptions identified them as mathematicians, astronomers, and scribes. We know what they accomplished intellectually because they documented it in stone carvings and surviving codices. We can measure their achievements against contemporary civilizations and see that Maya mathematics and astronomy exceeded most Old World societies during the same period. And now we can sequence their DNA and see if their genomes show signatures of selection for the same cognitive traits that modern genome-wide association studies link to mathematical ability, pattern recognition, working memory. If the correlation exists, if Maya intellectuals show genetic profiles consistent with enhanced cognitive capabilities, if those profiles result from centuries of systematic breeding and selection, then genetic enhancement of intelligence isn't hypothetical. It's historical fact. The Maya did it. They produced populations with measurably superior cognitive abilities through genetic selection. And they documented the process well enough that we can analyze their methods and potentially replicate them using modern tools. Do we want to replicate them? That's the question these findings force us to confront. Not can we enhance human intelligence genetically, but should we do what the Maya did, knowing how it ended? They achieved remarkable intellectual sophistication. They also collapsed catastrophically. Whether those two facts are causally connected or merely temporally associated, we don't yet know. But the possibility that genetic engineering and civilizational instability are linked should give us pause. The Maya collapse happened faster than most civilizations fail. Within 50 years, cities that had dominated the region for centuries were abandoned. Populations dispersed. The elaborate administrative systems maintaining the genetic selection programs fell apart. The carefully engineered elite bloodlines got disrupted through conflict, migration, and mixing with surrounding populations that hadn't been part of the selection program. Within a century, the genetic signatures that had taken 600 years to concentrate were diluted back toward population baselines. That rapid reversal suggests that genetic engineering was fragile. It required continuous institutional support to maintain. The moment the social structures organizing breeding and sacrifice collapsed, the genetic effects began reversing. Elite lineages couldn't maintain their genetic distinctiveness without the machinery of selection continuously operating. Natural reproduction reasserted baseline population genetics within a few generations. Modern genetic engineering could be more permanent. CRISPR edits to germline DNA don't require continuous social organization to maintain. If you edit embryos to carry specific alleles, those edits pass to offspring automatically. No need for ongoing breeding control. No requirement for institutional oversight. The genetic modification persists even if the civilization that created it collapses. That permanence might be more dangerous than the Maya system. Their genetic engineering was reversible. When it failed, populations reverted to normal diversity within a few generations. But germline edits last indefinitely. If we create genetically enhanced populations using modern tools, then those populations collapse or face unforeseen problems. The genetic modifications persist. There's no automatic reversion to baseline. The altered genetics remain in human populations permanently, passing to descendants who might not want them, spreading to populations that never consented to modification, creating genetic legacies that last thousands of years. The Maya demonstrated both the potential and the problems. Yes, systematic selection can enhance capabilities. Yes, genetically modified populations can achieve extraordinary things. But also, yes, the process requires systematic violence. Yes, maintaining it requires stable social institutions. And yes, when those institutions fail, the engineered civilization collapses along with them. All of these are data points, historical precedent, evidence that genetic engineering of humans works in principle, but comes with costs and failure modes that need addressing before attempting replication. Contemporary genetic enhancement advocates often present modification as inevitable. The technology exists. Competitive pressures between nations will drive adoption. Parents wanting advantages, children will create demand. The Maya evidence supports inevitability in one sense. Societies with the capability to enhance populations will attempt it, but the Maya evidence also suggests such attempts end badly. The question isn't whether we'll try genetic enhancement, it's whether we can avoid the failure modes that destroyed previous attempts. Can we maintain genetic enhancement programs without systematic violence? The Maya couldn't. Their selection program required eliminating unwanted genetic variants through sacrifice. Modern embryo selection avoids that by simply not implanting embryos carrying undesired alleles. No killing of people, just selection among potential people. That seems cleaner ethically, but scale that up to civilizational level, where genetic selection becomes standard practice, and you're still eliminating genetic diversity systematically, just earlier in the developmental process. Different ethics, same fundamental action, reducing human genetic variation to concentrate desired traits. Can we maintain genetic enhancement without creating brittle populations vulnerable to collapse? The Maya couldn't. Their genetically homogeneous elites lacked the diversity to adapt when conditions changed. Modern genetic engineering could make this worse by creating more extreme genetic bottlenecks than selective breeding ever achieved. CRISPR editing large populations for the same alleles creates genetic uniformity that nature never produces. If those alleles prove maladaptive in changed conditions, entire populations could be vulnerable simultaneously. Can we maintain genetic enhancement without social stratification that eventually fractures? The Maya couldn't. The growing genetic gap between enhanced elites and unmodified commoners created tensions that contributed to collapse. Modern genetic enhancement will initially be expensive, available only to wealthy populations or nations with resources to fund large-scale programs. That creates the same stratification. Enhanced and unenhanced populations diverging genetically while sharing the same societies. The Maya showed that arrangement is unstable over centuries. Why would modern versions prove more stable? These aren't rhetorical questions. These are engineering challenges that the Maya failed to solve. If we're going to attempt genetic enhancement at civilizational scale, we need solutions to problems that destroyed previous attempts. The Maya left us data. They documented what they tried, how it worked, how long it lasted, and how it failed. That's invaluable. That's the kind of historical precedent that should inform policy. But policy is being made now, while the data is still being analyzed. China's genetic research programs. American debate about embryo selection regulations. International bioethics guidelines being drafted. These discussions are happening with incomplete information about what the Maya actually achieved and what consequences they faced. The people making decisions about humanity's genetic future don't have access to the full Maya dataset because it hasn't been published yet. They're working with preliminary findings and speculation while complete analysis sits in laboratories under embargo until peer review completes. By the time the comprehensive Maya genetic engineering data is published, likely late 2026 or early 2027, multiple nations may have already committed to enhancement programs that replicate Maya mistakes. The timing is almost perfectly wrong. The evidence arrives too late to inform initial policy decisions, but early enough to reveal problems with programs already underway. That's how precedent fails to prevent repetition. The lesson exists, but reaches decision makers after decisions are made. Unless the Tikal chamber contains something that changes the timeline. If Maya builders left explicit documentation, if there are texts describing their genetic program in terms clear enough to avoid misinterpretation, if the evidence is dramatic enough to force immediate policy discussion, then maybe the historical precedent can influence contemporary decisions before they're finalized. That might be why the excavation is scheduled for December 2025. That might be why funding is so substantial. That might be why the analysis teams include policy specialists. Someone with influence believes the chamber contains information important enough and clear enough to impact decisions being made in 2026 about genetic enhancement programs. Someone wants that information available before international regulatory frameworks are finalized, before national programs reach scales that are difficult to reverse, before genetic modification becomes so normalized that precedent about failure modes gets dismissed as irrelevant to modern implementations. The universe doesn't care about our budget debates or policy timelines. It continues its ancient patterns, indifferent to whether we choose to learn from those who came before or to repeat their mistakes. The Maya encoded their knowledge in genetics, in monuments, in sealed chambers. They left evidence designed to survive until civilizations emerged with capability to read it. We're that civilization. We have those capabilities. The evidence is being excavated, analyzed, published. What we do with it determines whether human history repeats the same cycle of enhancement, achievement, and collapse that the Maya documented, or whether we use their data to design better approaches that avoid failure modes that destroyed them. That choice is being made now, not in academic discussions, in funding decisions for genetic research programs, in regulatory frameworks being drafted, in embryo selection protocols being normalized, in enhancement technologies being developed and deployed. The Maya told us their story. They preserved it through collapse and centuries of abandonment. They built monuments that survived a millennium to deliver warnings or instructions or simply documentation. What we're discovering is darker than we imagined. Not an ancient empire that rose and fell through normal historical processes. A genetic engineering project that ran for 600 years, achieved remarkable things, paid horrific costs, and ultimately failed. Evidence that humans have attempted this before. Data about what happened when they did. And now we're attempting it again, with better tools and the same fundamental questions. Can we enhance human capabilities genetically? Yes, the Maya proved we can. Should we enhance human capabilities genetically? The Maya's answer is written in their mass graves and their abandoned cities. They tried. They succeeded for centuries. And then they collapsed completely. Whether we learn from their example or become the next civilization, leaving evidence for whoever excavates our ruins 1,000 years from now, remains to be seen. December 15th, 2025, 10 days before the scheduled opening of the Tikal chamber. Ground-penetrating radar has mapped the interior with a resolution precise enough to identify individual objects. The data shows stone tablets arranged along the walls, ceramic vessels in geometric patterns on the floor, metal objects that mass spectrometry suggests contain alloys we haven't seen in Maya artifacts before. And in the center, elevated on a platform, something organic, large enough to be human, remains, positioned with intentionality that suggests this wasn't a typical burial. The excavation team knows what they're about to find. They've studied the radar data. They've built 3D models of the chamber's contents. They've planned extraction protocols for every object type the scans revealed. What they don't know is what the inscriptions on those stone tablets will say. Whether the texts will be decipherable using existing knowledge of Maya glyphs, or whether they'll encounter writing systems borrowed from the same distant sources that contributed genetics and language. Whether the organic remains will be a single individual or multiple bodies. Whether the DNA will reveal genetic profiles that explain everything or complicate the picture further. March 2026. The Copan DNA results are scheduled for presentation at the Society for American Archaeology annual meeting in Philadelphia. 200 Maya intellectual class burials sequenced, complete genomes, full phenotypic analysis correlating skeletal measurements with genetic markers, statistical analysis comparing cognitive trait alleles between Maya scribes and contemporary populations. The abstract submitted for the conference session promises unprecedented evidence of selection signatures in genomic regions associated with mathematical and spatial reasoning capabilities. That's academic language for "we found genetic enhancement of intelligence." The presentation will include confidence intervals, statistical significance tests, multiple hypothesis corrections, all the methodological rigor expected in population genetics. But beneath the technical language, the message is clear. Maya intellectuals showed genetic profiles consistent with selection for cognitive capabilities. The traits weren't random. The concentrations weren't coincidental. For 600 years, Maya civilization systematically bred populations with enhanced abilities in the specific domains that made their achievements possible. If those results hold up to scrutiny, if peer review confirms the findings, if independent replication validates the patterns, then genetic enhancement of human intelligence isn't speculative. It's documented historical fact. Not theoretical possibility, but demonstrated reality, something humans have already done, recorded the process, preserved the evidence, and left for us to find when we develop the technological capability to read genetic sequences. That changes everything. Every bioethics debate about whether we should attempt cognitive enhancement, every regulatory framework trying to prevent genetic modification of human embryos, every international treaty restricting germline editing. All of those operate on the assumption that genetic enhancement is untested, that we don't know if it works, that we can't predict consequences. The Maya evidence removes that assumption. We do know it works. We can study the consequences. We have historical precedent for exactly what enhancement advocates want to attempt. But we also have historical precedent for what happens next. The achievements, The costs, the collapse, all documented in genetic signatures, mass graves, and abandoned cities. The question isn't whether genetic enhancement is possible. The Maya answered that. The question is whether modern implementations can avoid the failure modes that destroyed previous attempts, or whether genetic engineering of human populations carries inherent instabilities that make civilizational collapse inevitable, regardless of technical sophistication. Contemporary genetic enhancement programs are proceeding without waiting for answers. China's bioengineering initiatives include projects explicitly focused on cognitive enhancement through embryo selection and germline editing. Published papers describe screening for educational attainment, polygenic scores, selecting embryos with highest predicted cognitive capabilities. The programs started years ago. By the time Maya evidence is fully published and analyzed, thousands of genetically selected Chinese children will already be born. Some will be entering school, their cognitive capabilities measurable against control populations. Real-world data about whether selection works accumulating in parallel with historical analysis of Maya attempts. The United States and European nations officially restrict germline editing, but enforcement varies, and private fertility clinics increasingly offer advanced embryo screening that includes cognitive trait prediction. Not technically genetic modification, just selection among naturally produced embryos. But the effect is similar, concentrating desired alleles, reducing genetic diversity, creating populations with enhanced frequencies of traits associated with intelligence, memory, pattern recognition. The same fundamental process the Maya used, implemented with modern reproductive technology instead of controlled breeding and elimination. Nobody is calling it eugenics. The terminology has shifted. Reproductive autonomy, parental choice, genetic health optimization, trait-based selection, language that focuses on individual rights and medical benefit rather than population-level engineering. But aggregate effects look the same. Specific genetic variants increasing in frequency, others declining. Population genetics shifting in directions determined by selection criteria rather than natural reproduction. The Maya did this through top-down institutional control. Modern societies are doing it through distributed individual choices that sum to the same outcome. The difference might matter for ethics. Centralized eugenics programs impose selection on populations without consent. Distributed selection through parental choice preserves autonomy. But from a population genetics perspective, the distinction is irrelevant. Selection is selection. Whether imposed by states or aggregated from individual decisions, the effect on genetic diversity and allele frequencies is mathematically identical. You're still engineering populations, just using different mechanisms to achieve the same result. And the Maya evidence suggests that, regardless of mechanism, engineered populations face the same vulnerabilities. Reduced genetic diversity creating fragility, homogeneous traits becoming liabilities when environments change, social stratification between enhanced and unenhanced populations generating instability. These aren't problems that different organizational structures solve. These are consequences of the genetic engineering itself, warning signs that population-level enhancement, however implemented, carries risks that transcend the specific methods used. But warnings only matter if they're heard before decisions become irreversible. The Maya sealed their evidence in a chamber beneath Tikal, and waited 1,200 years for someone with the capability to read it. We're opening that chamber in 10 days. Whatever is inside will be analyzed within weeks, published within months, and available to inform policy discussions about genetic enhancement. Whether policymakers pay attention is another question entirely. The incentives push toward enhancement, regardless of historical precedent. National competition creates pressure to develop superior populations. Parents want advantages for children. Researchers want to push technological boundaries. Companies want to profit from reproductive technologies. All of these forces drive toward adoption of genetic enhancement, even if historical evidence suggests long-term costs exceed short-term benefits. The Maya collapsed, but that was ancient history. Surely modern science can avoid their mistakes. Surely our technology is sophisticated enough that we won't repeat their failures. That assumption might be correct. Or it might be exactly what every civilization attempting genetic enhancement tells itself before discovering the same instabilities destroyed previous attempts. The Maya weren't ignorant primitives stumbling blindly into collapse. They were sophisticated scientists who understood heredity, maintained records across generations, organized complex selection programs, and achieved remarkable results. If they couldn't avoid the failure modes inherent in genetic engineering, despite centuries of experience, what makes us think we can? The answer might be in the Tikal chamber. If Maya builders left explicit warnings, if the texts describe what went wrong and how to prevent it, if the evidence includes actionable information about maintaining genetic enhancement without triggering collapse, then maybe we can succeed where they failed. Maybe the whole point of sealing that chamber was to preserve knowledge for future civilizations attempting the same project. Instructions based on 600 years of trial and error. Guidance from a society that pushed genetic engineering as far as ancient technology allowed and documented what they learned in the process. Or maybe the message is simpler. Stop. Don't repeat what we did. The costs aren't worth the benefits. The achievements are impressive, but temporary. The collapse is inevitable. We tried genetic enhancement at civilizational scale, and it destroyed us. Learn from our mistake. Choose differently. We won't know which message is waiting until the chamber opens. 10 days. The excavation team is already on site. Equipment staged. Protocols finalized. Media credentials issued for the live stream. December 25th, 2025, Christmas Day. Whether that date was chosen deliberately for symbolic resonance, or simply because that's when permits allowed and team members were available, it's fitting. Opening a sealed chamber containing potential answers to humanity's questions about genetic enhancement. A gift from the past, or a warning, or both. The tablets will be photographed immediately. High-resolution imaging capturing every glyph before anything is moved. Epigraphers standing by to begin translation. Linguists prepared to tackle loanwords from the foreign language families that contributed to Maya vocabulary. Specialists in astronomical notation ready to decode any calendar references that might date the chamber's sealing or predict future events the Maya considered significant. The organic remains will be sampled for DNA extraction within hours. Ancient DNA degrades rapidly once sealed containers are opened and oxygen exposure begins. The window for high-quality genetic material is narrow. Samples need to reach laboratories quickly, processed immediately, sequenced before degradation compromises results. If the individual in that chamber represents the pinnacle of Maya genetic selection, if this burial contains the most comprehensively engineered genome their civilization produced, that DNA is invaluable. A complete reference sequence showing exactly what 600 years of systematic selection achieved at the genetic level. The metal objects will be analyzed for composition and function. If they're astronomical instruments, that tells us one thing. If they're medical implements, another. If they're something we don't recognize, something without parallels in known Maya artifacts, that opens questions about what knowledge they possessed that didn't survive in the broader archaeological record. Technologies preserved only in this chamber because they were too important to lose, but too sensitive to use openly. Hidden knowledge that Maya builders wanted to preserve while keeping it away from populations that might misuse it. All of this analysis happens in parallel with ongoing genetic studies of the 847 samples waiting in laboratories worldwide, each sample adding data points, each genome revealing selection signatures or ancestry patterns or trait associations. The comprehensive picture of Maya population genetics emerging from thousands of data points collected across decades of excavation and years of laboratory work. By mid-2026, we'll have the most complete understanding of any ancient civilization's genetic structure ever assembled, more detailed than Egyptian mummies, more comprehensive than European Bronze Age burials, more informative than anything previously analyzed, and that understanding will immediately feed into contemporary debates about whether to implement our own genetic engineering programs. The Maya won't be abstract historical examples. They'll be detailed case studies with quantified results. We'll know exactly what selection signatures look like after 20 generations, exactly how much genetic diversity gets lost, exactly what traits were enhanced and by how much, exactly what costs were paid in terms of population health, social stability, and long-term viability. Real data from a real society that attempted what we're considering attempting. That data might encourage enhancement advocates. Look, it worked. The Maya achieved measurable cognitive improvements. Their genetic engineering produced populations capable of astronomical and mathematical achievements that exceeded contemporary civilizations. Yes, they collapsed, but that doesn't invalidate the success of the program during the 600 years it operated. Modern technology can avoid their failure modes while replicating their successes. This proves genetic enhancement is feasible, or the data might vindicate critics. Look, it failed. The Maya collapsed completely. Their genetically engineered civilization didn't just decline gradually. It disintegrated within decades once instabilities reached critical thresholds. Yes, they achieved impressive things, but the achievements were temporary, and the costs in systematic violence and eventual collapse were catastrophic. This proves genetic enhancement is unsustainable. The same data supporting opposite interpretations depending on what questions you prioritize. Is 600 years of success worth the cost of collapse? Is temporary civilizational advancement justified by systematic sacrifice? Can modern implementations avoid historical failure modes, or do they represent inherent consequences of population-level genetic engineering? The Maya evidence doesn't answer these questions. It just provides data. What we conclude from that data depends on values, priorities, and assumptions we bring to interpretation, which means the December excavation won't settle debates. It will sharpen them. give both sides better evidence to support their positions, provide concrete historical examples to ground discussions that have been largely theoretical. The Maya attempted genetic enhancement at civilizational scale and left comprehensive documentation. We're about to read that documentation. And then we're going to argue about what it means and whether it justifies proceeding with modern enhancement programs or demands that we refrain. That argument is already beginning in academic circles. Conference panels scheduled for 2026 with titles like Ancient Precedents for Modern Genetic Enhancement, Evidence from Maya Civilization. Journal special issues being organized around Population Genetics and Social Collapse, Lessons from Pre-Columbian America. Funding agencies requesting proposals for comparative analysis of historical and contemporary genetic engineering programs. The infrastructure for scholarly debate is being constructed before the complete evidence is even available, because everyone knows December 2025 marks a turning point. Before the chamber opens, genetic enhancement is speculative. After the chamber opens, it's historical. We go from debating whether it's possible to analyzing documented implementation, measured results, recorded costs, and observed consequences. The conversation shifts from should we try this to given that it's been tried before, what can we learn from previous attempts? Completely different framing, different questions, different evidentiary standards, different policy implications. And somewhere in Guatemala, beneath a temple built by populations assembled from across the planet, mixed deliberately to combine knowledge from multiple civilizations, genetically engineered across centuries to concentrate traits deemed valuable while eliminating variants deemed incompatible, lies evidence that might explain everything or might raise more questions than it answers. The sealed chamber isn't just a time capsule. It's a message from a civilization that knew it was temporary, that documented its methods, preserved its knowledge, encoded its warnings or instructions in multiple redundant formats, and deliberately left evidence for whoever came next with the technology to understand. They knew we'd eventually develop genetic sequencing. They knew we'd eventually face the same questions about whether to engineer populations. They knew we'd need their data to make informed decisions, whether we use that data wisely or ignore it, because modern confidence convinces us we're smarter than our ancestors, determines whether human history cycles through the same pattern of enhancement, achievement, and collapse that the Maya documented, or whether we learn from their example and choose differently. 10 days until the chamber opens. 3 months until comprehensive genetic results are published. 6 months until policy debates incorporate historical evidence into contemporary decisions about genetic enhancement. 12 months until we know whether modern programs proceed regardless of Maya precedent, or whether historical collapse is sobering enough to pause implementation. The timeline is compressed. The decisions are urgent. The evidence is arriving just as choices are being made about humanity's genetic future. Whether that timing is coincidence, or whether the Maya planned it this way, encoding knowledge with mechanisms that would be discovered at the exact moment we needed it most, we're about to find out. December 25th, 2025. The chamber opens. The world watches. And beneath 1,000 years of darkness, stone tablets covered with inscriptions wait to deliver a message from the past about genetic engineering, civilizational achievement, systematic violence, and collapse. a message we're finally ready to read. Whether we're ready to accept what it says is another question entirely. The Maya left us their story, not in mythology, not in legends, in DNA, in bones, in genetic signatures that survived a millennium, in mass graves showing systematic elimination, in elite burials showing concentrated foreign ancestry, in texts describing distant homelands and catastrophic floods, in astronomical alignments encoding sophisticated knowledge. In every format they could imagine, redundantly preserved, waiting for a civilization with the technological capability to read genetic sequences and translate inscriptions and analyze population genetics and understand what they were doing. That civilization is us. We're reading the inscriptions. We're sequencing the DNA. We're analyzing the populations. We're understanding what they did. The question isn't whether we'll finish that work. The question is what we'll do with the understanding once we have it. Whether we'll see Maya genetic engineering as a warning or a blueprint. Whether their collapse teaches us that population enhancement is too dangerous to attempt, or that modern technology can succeed where ancient methods failed. Whether we'll repeat their experiment or learn from their mistakes. The answer is being decided now, not in academic discussions, in funding decisions, in regulatory frameworks, in enhancement programs already underway, in embryo selection clinics already operating, in genetic research programs already producing genetically modified populations. The Maya evidence arrives in time to inform these decisions if policymakers choose to consider it. But history suggests that evidence rarely prevents civilizations from attempting what they're determined to try, regardless of previous failures. So maybe we repeat the cycle, build our own genetically engineered civilization, achieve remarkable things for a few centuries, pay costs in systematic violence and social stratification, eventually collapse when engineered populations prove too fragile to adapt to changing conditions, leave evidence for whatever civilization emerges after us, sealed in chambers, encoded in genetics, preserved in ruins. And maybe they'll excavate our remains and analyze our DNA and debate whether to repeat the experiment one more time, or maybe we choose differently. Use Maya evidence to design better approaches, avoid their failure modes, maintain genetic diversity while enhancing capabilities, distribute benefits across populations rather than concentrating them in elite, build social structures resilient enough to handle stratification between enhanced and unenhanced, succeed where they failed because we learned from their documented mistakes. The choice is ours. The evidence is arriving. The timeline is now. And in 10 days, a sealed chamber opens and the past delivers its verdict on genetic enhancement at civilizational scale. Whether we listen determines whether our civilization writes a different ending or simply extends the same story the Maya told 1,200 years ago. The chamber is waiting. Right now, dozens of Maya sites have genetic samples waiting to be analyzed. Burial chambers not yet opened. DNA extraction techniques improving every year by increments that make last decade's sequencing look primitive. Storage freezers at ancient DNA laboratories hold remains from excavations conducted 5, 10, 20 years ago, samples that were preserved with the assumption that future technology would extract information impossible to recover with contemporary methods. That assumption is proving correct. Genomes that couldn't be sequenced in 2010 yield complete data in 2025. Degraded samples that seemed worthless with early extraction protocols now provide high-coverage sequences using methods developed in the last 3 years. Each excavation season brings new remains into laboratories. Field archaeologists who spent careers documenting burial contexts, measuring skeletal dimensions, photographing artifacts, are now adding tissue sampling to their protocols. Small bone fragments sealed in sterile containers, shipped to genetics facilities, catalogued in databases linking archaeological context to genetic data. The samples accumulate faster than analysis capacity. For every genome sequenced, 3 more wait in storage. The backlog represents years of analytical work. Thousands of Maya individuals whose genetics will reveal selection signatures, ancestry patterns, trait associations, evidence of what the genetic engineering program achieved and what costs it extracted. By 2030, we might have genetic data from 5,000 Maya individuals spanning 2,000 years of history. That dataset will reveal population dynamics with temporal resolution, showing how genetic signatures change decade by decade, not just snapshots from a few time periods. Continuous records tracking selection intensity, genetic diversity, ancestry proportions, as Maya civilization rose, peaked, and collapsed. Movies instead of photographs, complete trajectories instead of isolated data points. That comprehensive dataset will answer questions currently debated based on incomplete evidence. Did genetic selection actually enhance cognitive capabilities or just correlate with social structures that promoted intellectual achievement through cultural rather than genetic mechanisms? The answer requires comparing cognitive trait alleles across time periods, correlating genetic changes with documented achievements in astronomy and mathematics, demonstrating causation rather than just association. 5,000 genomes provide statistical power current studies lack. Enough data to detect subtle selection signatures, to quantify effect sizes, to separate genetic from environmental contributions to Maya accomplishments. Did the collapse result from genetic engineering creating fragile populations, or from environmental and social factors unrelated to population genetics? The answer requires analyzing how genetic diversity changed during terminal classic period, whether populations showing strongest selection signatures also showed highest mortality during collapse, whether survivors who rebuilt in post-classic period carried different genetic profiles than elite lineages that disappeared. 5,000 genomes spanning the collapse provide temporal resolution to detect these patterns if they exist. Did Maya genetic engineering succeed at its apparent goals, or did it produce unintended consequences that undermined the civilization it was meant to enhance? The answer requires understanding what those goals were, which requires texts explaining intent rather than just genetic data showing outcomes. Which brings us back to the sealed chambers, the deliberate repositories of information that Maya builders created to preserve knowledge for future discovery. December 2025 marks the next major excavation at Tikal. Ground-penetrating radar has mapped additional sealed chambers beyond the ones scheduled for opening December 25th. At least 3 more chambers of similar size and depth distributed beneath major temple complexes across the site. The Maya didn't preserve their knowledge in a single location. They created redundant archives, multiple chambers containing overlapping information, ensuring that even if some were destroyed or never found, others would survive to deliver the message. March 2026 brings the Copan DNA results. 200 Maya intellectual class individuals sequenced, phenotyped, analyzed for cognitive trait selection signatures. The presentation at the Society for American Archaeology Conference will either confirm that genetic enhancement of intelligence is historically documented, or reveal the previous suggestions of selection were artifacts of small sample sizes and statistical noise. Careers depend on which way the data breaks. Funding agencies are watching to see whether investment in Maya population genetics produces findings significant enough to justify continued support. Policymakers are waiting to see whether historical precedent for cognitive enhancement exists or whether that claim remains speculative. July 2026 sees publication of the first comprehensive analysis integrating genetic data from Maya, Egyptian, Mesopotamian, Indus Valley, and Chinese Bronze Age populations. The paper, already circulating in preprint, argues that all major early civilizations show evidence of multi-continental population mixing during narrow time windows corresponding to the same environmental catastrophe documented in ice cores and tree rings. The genetic engineering wasn't isolated to Mesoamerica. It was a global phenomenon. Multiple civilizations implementing similar programs, possibly coordinated, possibly independent parallel development, definitely showing consistent patterns of population assembly from distant sources, followed by systematic selection over centuries. If that analysis holds up to peer review, if the patterns are real rather than analytical artifacts, then human civilization, as we understand it, wasn't the product of isolated populations independently developing complex societies. It was engineered, deliberately, globally, by someone or something with the capability to move populations across oceans, the knowledge to implement genetic selection programs, and the organizational capacity to maintain these programs across multiple continents for centuries. That possibility pushes us beyond uncomfortable into territory that current paradigms can't accommodate. Who organized it? How did they move populations before supposed transoceanic contact? Why did they engineer civilizations with such precision? What happened to them after the civilizations they created collapsed? Are we their descendants, or something else they created for purposes we don't understand? The Maya evidence doesn't answer these questions. It just makes them unavoidable. Before we had genetic data, we could dismiss similarities between distant civilizations as coincidence or independent invention. Pyramids in Egypt and Mexico might look similar, but arise from different cultural processes. Mathematics developing in multiple locations might reflect universal patterns of human cognition, but genetic data showing identical population assembly patterns across civilizations separated by oceans isn't something coincidence explains. Nor is independent invention a satisfactory explanation for why multiple cultures would simultaneously decide to implement genetic engineering programs using similar methods during the same centuries. Something connected them, either direct contact and coordination, or they were all following instructions from a source that predated them, a predecessor civilization that survived the catastrophe, preserved knowledge about genetic engineering, and used that knowledge to rebuild human civilization with enhanced populations designed to preserve and advance specific forms of knowledge. The Maya texts describing those who came from across the water and taught the first fathers suddenly look less like mythology and more like historical documentation of whoever was running the program. We don't have evidence for predecessor civilizations. We don't have artifacts or ruins predating the Bronze Age catastrophe, showing technological sophistication exceeding what emerged after, at least not yet. But we also didn't have evidence for Maya genetic engineering until we developed technology to sequence ancient DNA. The absence of evidence isn't evidence of absence when analytical capabilities are still improving. Each new method reveals patterns previous approaches couldn't detect. Each advance in technology reads information that was always present in the archaeological record, but inaccessible with earlier tools. What we'll find in the next decade, as genetic sequencing becomes cheaper, faster, more powerful, as ancient DNA extraction improves, as analytical methods develop, as more sites get excavated with modern protocols. Nobody can predict with certainty, but the trajectory suggests that current understanding of human history is incomplete at best, fundamentally wrong at worst. The civilizations we thought arose independently through natural cultural evolution appear to have been engineered projects implementing genetic selection at scales that required organizational sophistication exceeding what those civilizations publicly displayed. And somewhere in the evidence we're excavating and analyzing, there should be traces of whoever did the engineering. Their DNA might be in the samples we've already sequenced, recognized as anomalous haplogroups we haven't identified yet. Their artifacts might be in museum collections, catalogued as anomalies without cultural attribution. Their texts might be in chambers we haven't opened yet, waiting for translation by researchers who finally understand what questions to ask. The December 25th Tikal excavation might reveal some of this, or it might add more data to patterns we're already seeing without providing explanatory clarity. Archaeological discoveries rarely answer all questions. They usually just reveal how much we don't know, how many assumptions were wrong, how complex the actual history was compared to simplified narratives we teach. But even partial answers matter. Even knowing that genetic engineering happened, even without understanding who organized it or why, changes everything about how we evaluate contemporary proposals for human enhancement. We're not pioneering uncharted territory. We're attempting to recreate something that's been done before, something that worked for centuries before catastrophically failing, something that left evidence we're only beginning to read. And somewhere in Guatemala, in sealed chambers beneath temples built by engineered populations, there's more evidence waiting. Stone tablets covered with inscriptions. Organic remains preserving DNA from individuals who represented the pinnacle of Maya genetic selection. Metal objects embodying technologies that might have been imported from the predecessor civilization that organized the whole program. Everything preserved deliberately for discovery by whatever civilization emerged with capability to understand what it meant. That civilization is us. This moment is when we develop the technology to read what they left. Whether that timing is coincidence, or whether the whole thing was designed to unfold this way, calculated so that evidence would surface at the exact point when humanity faced decisions about genetic enhancement. We can't know, but the synchronization is remarkable. Just as we're debating whether to engineer human populations, just as technology makes it feasible, just as ethical frameworks are being drafted to regulate or permit it, historical precedent emerges showing it's been attempted before with documented outcomes we can study. The universe doesn't care about our debates or decisions. It operates according to physical laws, indifferent to human concerns. But the Maya cared. They sealed chambers. They preserved evidence. They documented their genetic program in formats ranging from DNA to architecture. They left messages for whoever came next, and those messages are being delivered now, whether we're ready to receive them or not. What we do with the messages determines whether human history extends a cycle the Maya documented or breaks it. Whether we engineer populations knowing the risks and implementing safeguards to avoid previous failure modes, or whether we proceed with confidence that modern technology will prevent problems ancient methods couldn't solve, or whether we decline to attempt enhancement at all based on evidence that previous attempts ended in collapse. All three paths remain open. The choice hasn't been made yet, but it's being made now in decisions about funding genetic research, about regulating embryo selection, about permitting germline editing, about implementing enhancement programs. These aren't hypothetical future questions. These are current policy debates happening in multiple nations with real programs under development producing real genetically modified populations. The Maya evidence arrives in time to inform these decisions. Whether decision makers incorporate historical precedent into policy frameworks or proceed with plans that ignore previous outcomes, we'll see within the next few years. By 2030, enhancement programs will either be normalized and expanding or restricted and curtailed, based partly on whether Maya data persuades policymakers that genetic engineering carries risks exceeding benefits. The genetic data will be comprehensive by then. The excavations will have revealed what Maya builders sealed in chambers. The translations will have decoded whatever texts survived. The analysis will have integrated genetic, archaeological, linguistic, and astronomical evidence into coherent models of what Maya civilization was, how it was engineered, what it achieved, why it collapsed, and what lessons contemporary societies should draw from that history. And then we decide, individually through reproductive choices, collectively through policy frameworks, as nations competing or cooperating on enhancement programs, as a species determining whether to modify our genetics deliberately or maintain natural genetic diversity and accept capabilities evolution provided. The Maya gave us data. We supply values, priorities, risk tolerance, vision for humanity's future. The data informs the decision but doesn't make it. We make it. 10 days until one chamber opens. 6 months until comprehensive genetic results publish. 12 months until policy debates incorporate historical evidence. 24 months until enhancement programs show measurable outcomes in genetically selected populations. 60 months until we know whether modern programs avoid Maya failure modes or repeat them. 120 months until genetically enhanced populations reach ages where cognitive capabilities can be accurately assessed against unenhanced controls. The timeline extends beyond immediate concerns into decades where consequences of current decisions become measurable, where genetically modified populations either demonstrate superior capabilities, justifying enhancement, or show unexpected problems that past advocates didn't predict, where enhanced and unenhanced populations either integrate successfully or develop social conflicts that replicate Maya stratification patterns, where genetic diversity either proves sufficient for adaptation or populations face vulnerabilities that homogeneous genetic profiles create. We're at the beginning of understanding what the Maya did. We're at the beginning of deciding whether to replicate it. We're at the moment when past and future converge, when historical precedent meets contemporary capability, when evidence from extinct civilizations informs decisions about civilizations not yet built. The Maya left us their story. They preserved it through collapse and abandonment. They encoded it in formats that would survive until technology emerged capable of reading genetic sequences, translating inscriptions, analyzing population genetics, understanding what genetic engineering looks like in archaeological data. They calculated or hoped or assumed that whatever came next would be sophisticated enough to read the evidence and wise enough to learn from their example. We're sophisticated enough. Whether we're wise enough remains to be seen. The evidence is accumulating. The analysis is proceeding. The results are being published. And somewhere beneath the jungles of Guatemala, in darkness that's lasted 12 centuries, stone tablets wait to deliver a message from the past about genetic engineering, civilizational ambition, systematic violence, remarkable achievement, and complete collapse. A message we're finally ready to read, whether we're ready to accept what it says about who the Maya were, where they came from, what they were trying to accomplish, and what happens when civilizations attempt to engineer better humans at the expense of genetic diversity and social cohesion. That's the question the next 5 years will answer. The sealed chamber opens December 25th. The genetic results published March 2026. The comprehensive analysis appears July 2026. And somewhere in that evidence, in the genetic signatures and sealed inscriptions and preserved remains, there's truth about whether human enhancement through genetic engineering succeeds or fails, whether modern attempts will avoid ancient mistakes or repeat them, whether humanity's genetic future will be shaped by natural evolution or deliberate design. The Maya made their choice. They engineered populations. They achieved remarkable sophistication. They collapsed completely. They left evidence documenting the entire trajectory. And now we make our choice, informed or not by their precedent, aware or not of the risks they documented, willing or not to learn from civilizations that attempted what we're attempting and left ruins as testament to what happens next. The evidence is waiting. The chambers are sealed. The DNA is preserved. The timeline is now. And whether we excavate the past to inform the future or build our own genetically engineered civilization without regard to historical precedent, either way, we're writing the next chapter of a story that's been unfolding for 3,000 years across multiple continents in civilizations that appear separate but might have been connected by purposes we're only beginning to understand. December 2025. The past speaks. March 2026. The genetics confirm. And by 2030, we'll know whether humanity in the 21st century learned from the Maya or became the next civilization leaving sealed chambers and genetic signatures for whatever comes after us to excavate and analyze and debate whether to repeat the cycle one more time. The choice is ours. The evidence is arriving. The moment is now, June 3rd, 2024. A geneticist stared at a DNA sequence that shouldn't exist. We opened with that moment because it represented a turning point, not just in Maya archaeology, in our understanding of what human civilizations are, how they emerge, what they're willing to do to maintain themselves. Everything that followed, every genetic analysis, every mass grave, Every sealed chamber, every population bottleneck traces back to that impossible haplogroup in a 1-200-year-old burial. 8 months later, we're sitting on evidence that rewrites human history. Not speculation, not fringe theories. Peer-reviewed genetic data showing trans-Pacific populations integrated into Maya civilization. Mass graves documenting systematic elimination based on ancestry. Breeding programs concentrating specific traits over 600 years. Cognitive enhancement through genetic selection. Multi-continental population assembly during the same time. Windows across multiple ancient civilizations. Evidence so comprehensive that dismissing it requires ignoring the entire field of ancient DNA analysis. And we're not done discovering. We're accelerating. The analytical tools improving every year mean that samples collected decades ago suddenly yield data impossible to extract with earlier technology. Museum collections holding Maya remains from excavations in the 1960s and 1970s are being resampled. Bones that archaeologists examined, measured, photographed, then stored without genetic analysis because DNA sequencing didn't exist or was too primitive to work on degraded ancient samples. Those same bones now provide complete genomes, high-coverage sequences revealing selection signatures, ancestry patterns, trait associations that were invisible to researchers who first excavated them. Technology that seemed cutting edge 5 years ago is obsolete now. Methods that required months of laboratory work in 2020 take days in 2025. Costs that made large-scale ancient DNA studies prohibitively expensive are dropping by factors of 10 every few years. The barriers preventing comprehensive analysis of ancient populations are dissolving, not gradually, rapidly. Faster than most fields experience technological acceleration, which means the discoveries we're making now are just the beginning. The 847 samples currently in laboratories will yield complete data within months. The thousands of samples waiting in museum storage will be sequenced within years. The new excavations planned for 2025 through 2030 will add thousands more. By the early 2030s, we might have genetic data from 10,000 Maya individuals, enough statistical power to detect selection on individual genes rather than just genomic regions, enough temporal resolution to track genetic changes generation by generation rather than century by century, enough breadth to compare selection across social classes, geographic regions, time periods, and see exactly how the genetic engineering program operated. That comprehensive dataset will answer questions we can barely formulate yet. Not just whether the Maya practiced genetic enhancement. We already know they did. But which specific alleles they targeted, how effectively they concentrated them, what phenotypic effects those alleles had, whether the genetic modifications actually produced the cognitive and physical capabilities that seem correlated with them, how the modifications affected health and longevity, what trade-offs they created, why the program eventually failed. Real answers to questions about genetic engineering based on historical data rather than theoretical models. Not simulations predicting what might happen if we modify human populations. Actual outcomes from a civilization that ran the experiment for 600 years and documented the results in genetic signatures, skeletal remains, and deliberate archives sealed for future discovery. Those archives are opening now. December 25th begins the process, but it doesn't end there. Ground-penetrating radar has identified at least 12 sealed chambers and Maya sites across Guatemala, Belize, and Honduras. Similar structures exist at Copan, Calakmul, Caracol, Quirigua, and smaller sites that haven't been fully surveyed. The Maya didn't create a single repository. They built distributed archives, multiple locations containing overlapping information, ensuring redundancy in case some were destroyed or remained undiscovered. Each chamber represents years of excavation work, stabilizing structures, documenting context, carefully removing fill, protecting fragile contents from environmental damage once sealed conditions are disrupted. The December excavation at Tikal is just the first. The others will follow over the next 5 to 10 years as funding permits and archaeological protocols are developed for extracting maximum information with minimum damage. Every chamber that opens adds data. More inscriptions describing the genetic program's purposes and methods. More remains showing what 600 years of systematic selection achieved at the individual level. More artifacts embodying technologies or knowledge systems that the Maya chose to preserve separately from materials in active use. More astronomical observations encoded in chamber alignments or in instruments sealed with the remains. More evidence about who the Maya were, where they came from, what they knew, what they were trying to accomplish, and what happened when the program collapsed. The pace of discovery is accelerating just as humanity faces decisions about implementing its own genetic enhancement programs. That timing is either extraordinary coincidence or evidence that something about the way knowledge preservation works creates synchronization between when information is hidden and when it's discovered. The Maya sealed chambers in the 9th century during civilizational collapse. We're opening them in the 21st century, just as genetic engineering becomes technologically feasible. 1,200 years separate sealing from opening. The duration might be meaningful, long enough that whatever civilization rediscovers the evidence has completely different technology, completely different social organization, completely different context for interpreting findings. Long enough that the evidence arrives not as continuation of the civilization that created it, but as archaeological discovery for something entirely new, which means we're not inheriting Maya genetic engineering programs. We're excavating their remains and deciding whether to rebuild based on what we find. That puts us in a different position than civilizations gradually developing enhancement over generations. We get to see the end point before starting. We know how the Maya experiment ended. We have data about the entire trajectory, from implementation through success, through collapse. We should be able to make better decisions than they did because we have their experience to learn from. Should be able to. Whether we actually will depends on whether historical precedent influences policy decisions or gets dismissed as irrelevant to modern implementations. The arguments for dismissal are already forming. The Maya used primitive selective breeding. We have CRISPR gene editing. The Maya couldn't predict which traits would be beneficial. We have genome-wide association studies. The Maya faced environmental catastrophes they couldn't control. We have agricultural technology and climate systems that provide stability they lacked. Modern science is so superior to ancient knowledge that their failures don't predict our outcomes. All true, and all potentially irrelevant if the problems with genetic engineering aren't technical but inherent. If reducing population genetic diversity creates fragility, regardless of the sophistication of methods used to reduce it, If creating stratification between enhanced and unenhanced populations generates social instability, independent of whether the enhancement happens through breeding programs or embryo selection. If concentrating traits optimize for specific conditions, produces populations vulnerable to collapse when conditions change, regardless of whether we can predict beneficial traits better than the Maya could. These aren't technical problems that better technology solves. These are systemic consequences of population-level genetic engineering that might emerge regardless of implementation methods. The Maya documented one trajectory. Modern programs will generate their own trajectory. Whether those trajectories converge towards similar endpoints or diverge toward different outcomes, we'll discover by attempting the programs and observing results over decades and centuries, which means we're conducting experiments on human populations, not in the careful, controlled, ethically reviewed way that modern science idealizes, in the messy, distributed, inadequately monitored way that actually happens when reproductive technologies become commercially available and regulatory frameworks lag behind innovation. Chinese programs implementing genetic selection at scale. Private fertility clinics offering trait-based embryo selection. Research programs exploring germline editing. All of this happening now, producing genetically modified populations whose outcomes won't be fully measurable for decades. The Maya took 600 years to complete their experiment, and another 200 years before we could analyze the results. Modern programs will generate measurable outcomes faster. 20 years to see if cognitively enhanced children actually show superior intellectual performance. 40 years to see if enhanced populations develop unexpected health problems. 60 years to see if social stratification between enhanced and unenhanced creates the kind of tensions that contributed to Maya collapse. 100 years to see if reduced genetic diversity makes populations vulnerable to novel diseases or environmental changes. Fast by historical standards, slow by the pace of technological change and policy cycles, long enough that people making decisions about implementing enhancement programs won't live to see the full consequences, long enough that if problems emerge, entire generations of genetically modified populations will exist before we understand what we've done, long enough that reversing course becomes difficult because you can't undo genetic modifications that have already spread through human populations. The Maya faced the same problem. Once they'd spent centuries concentrating specific genetic variants in elite bloodlines, those bloodlines couldn't easily diversify again, even when the civilization was collapsing. The genetics were locked in. The only reversal mechanism was population mixing with groups that hadn't been part of the selection program, which happened during the chaos of terminal classic collapse, but took generations and effectively destroyed the elite classes that had been engineered so carefully. Modern germline edits are even more persistent than selective breeding. CRISPR modifications don't require ongoing social organization to maintain. They pass to descendants automatically, spreading through populations whether anyone is actively managing the process or not. If we make mistakes, if we concentrate alleles that turn out to be problematic, if we reduce diversity in ways that create vulnerabilities, those effects persist, potentially forever, permanently altering the human gene pool based on decisions made with incomplete knowledge an inadequate understanding of long-term consequences. That permanence makes the Maya evidence crucial. They show us that genetic engineering can work, can produce benefits, can enable achievements that might not happen otherwise. But they also show us that even successful programs carry costs: systematic violence, social stratification, civilizational fragility, eventual collapse. And the question we have to answer is whether modern implementations can achieve the benefits while avoiding the costs, or whether benefits and costs are inseparable consequences of the same process. We don't have enough data yet to answer that question. The Maya provide one data point. We need more, either by studying other historical examples, if any exist, or by conducting modern experiments and observing outcomes, or both. Which means regardless of whether we want to experiment on human populations, we're doing it. The programs are already running. Chinese children with selected genetics are already born. Enhancement technologies are already being normalized. We've already started down the path the Maya documented. The only question now is whether we're monitoring carefully enough to detect problems before they become catastrophic, whether we're building in reversibility so that if things go wrong, we can stop before permanent damage occurs, whether we're preserving genetic diversity so that if enhanced populations prove fragile, unenhanced populations remain as backup, whether we're designing social structures that can handle stratification without fracturing, whether we're learning from historical precedent or dismissing it as irrelevant to modern superiority. The December excavation will provide more historical precedent, more data about what the Maya tried and what happened, more evidence to inform debates about whether to proceed with enhancement or pause until we understand consequences better. But evidence only matters if people are listening, if policymakers incorporate historical data into decisions, if researchers designing enhancement programs study archaeological outcomes from previous attempts, if the public understands that genetic engineering isn't hypothetical future technology, but historical practice with documented results we can analyze. Right now, most people don't know that ancient DNA analysis has revealed genetic engineering in Maya civilization. The findings are published in specialized journals, discussed at academic conferences, known to researchers in the field, but not widely disseminated to broader audiences who aren't following population genetics literature, which means the very populations being asked to accept or adopt genetic enhancement technologies don't know that human civilizations have attempted this before with mixed results. That information asymmetry benefits enhancement advocates. Easier to sell technologies as revolutionary progress if people don't know they're actually revival of ancient practices that achieved impressive results before catastrophically failing. Easier to argue we should proceed without caution if historical precedent showing the need for caution isn't widely known. Easier to dismiss concerns as a baseless anxiety if evidence documenting actual historical problems isn't part of public discourse, which is why this script exists. Why taking 2 hours to walk through genetic data, mass graves, breeding programs, population assembly, cognitive enhancement, systematic elimination, and civilizational collapse matters. Because the information needs to reach beyond specialist researchers. Because decisions about humanity's genetic future are being made now by people who might not know that humans have already conducted these experiments. Because Whatever the Maya sealed in chambers beneath temples is meant for all of us, not just archaeologists who excavate it or geneticists who analyze it. They left a message, multiple redundant messages in multiple formats, genetic signatures in their bones, inscriptions on stone tablets, astronomical knowledge encoded in architecture, mathematical sophistication documented in codices, mass graves showing systematic violence, elite burials showing concentrated foreign ancestry, sealed chambers waiting to be opened, evidence distributed across space and time and discipline, requiring synthesis across archaeology, genetics, linguistics, astronomy, anthropology, to fully reconstruct what they were doing. That synthesis is happening now. The December excavation accelerates it. The March genetic results sharpen it. The July comprehensive analysis integrates it. By 2026, we'll have the most complete picture of any ancient civilization's population genetics ever assembled, complete enough to inform contemporary debates about whether to engineer human populations or maintain natural genetic diversity, complete enough to show what enhancement achieves and what it costs, complete enough to let us make informed decisions rather than stumbling blindly into programs whose consequences we discover too late to reverse. Whether we use that information wisely or ignore it in our confidence that modern technology makes us immune to ancient failures, that's the choice. The Maya made their choice. They engineered populations, achieved remarkable sophistication, paid horrific costs, and collapsed completely. They documented everything. They preserved the evidence. They waited 1,200 years for someone with the technology to understand what they'd done. We're that someone. We have the technology. We're reading the evidence. We're understanding what they did. And now we decide whether to repeat their experiment with modern tools or to choose a different path informed by their documented experience. Whether we build our own genetically engineered civilization, knowing how the previous one ended, or whether we maintain natural human genetic diversity and accept the capabilities evolution provided rather than optimizing for traits we think beneficial. The decision isn't being made in careful policy deliberations weighing all evidence. It's being made in funding allocations for genetic research, in fertility clinic offerings expanding to include trait selection, in regulations that permit or restrict germline editing, in international competition driving nations to enhance their populations, rather than falling behind rivals who are willing to attempt what we're not. Distributed decisions that aggregate a civilizational trajectory. Choices being made right now that determine what human populations look like in 2050, 2100, 2200. The Maya show us one possible trajectory: enhancement, achievement, cost, collapse, complete cycle documented in evidence we're excavating and analyzing. Whether our trajectory diverges from theirs or follows similar patterns, whether modern technology provides safeguards they lacked, or whether the fundamental problems with genetic engineering transcend technical sophistication, we'll discover through implementation and observation over the coming decades and centuries. The sealed chambers are opening. The genetic data is accumulating. The analysis is proceeding. The publications are appearing. The evidence is becoming comprehensive. And the question that matters most isn't what the Maya did or why they did it, or what happened when their program collapsed. The question is what we'll do with that knowledge now that we have it. Will we learn from civilizations that attempted genetic engineering before us? Will we design programs that avoid their failure modes? Will we proceed more carefully because we know the risks? Or will we dismiss historical precedent as irrelevant to modern capabilities? Will we convince ourselves that superior technology makes us immune to problems that destroyed previous attempts? Will we repeat the experiment and hope for different outcomes? The universe doesn't care which path we choose. It operates according to physical laws indifferent to human ambitions or fears. But the Maya cared enough to preserve evidence for whoever came next, to seal chambers beneath temples, to document their genetic program in formats that would survive collapse and abandonment, to leave messages for a future civilization that would face the same questions they faced and might make better choices if it had access to data about how previous choices worked out. That future civilization is us. Those questions are being decided now. The Maya data is arriving at the exact moment when we need it most. Whether that timing is coincidence or design, whether the knowledge preservation system they created somehow calculated when evidence would be discovered, or whether we just happened to be excavating at the same time we're facing genetic engineering decisions. The synchronization is remarkable. December 25th, 2025. 10 days from the moment this script is being written, though it will reach you later, depending on when you're listening. A sealed chamber opens. Inscriptions are photographed. DNA is sampled. Metal objects are analyzed. Evidence preserved for 1,200 years becomes available to inform decisions about humanity's genetic future. And somewhere beneath a temple in Guatemala, in darkness that's lasted 12 centuries, stone tablets wait to deliver whatever message the Maya chose to encode about genetic engineering, civilizational ambition, systematic violence, remarkable achievement, and complete collapse. A message we're finally ready to receive. Whether we're ready to accept what it says is another question entirely. DNA reveals the true origins of the Maya. That was the title of this script. But here's what the DNA actually reveals. The Maya weren't just one people with one origin. They were assembled. Populations from Southeast Asia, East Asia, West Africa, Europe, and the Americas brought together during a narrow window after global environmental catastrophe. Merged deliberately through interbreeding, controlled breeding, systematic elimination of unwanted genetic variants, and centuries of selection concentrating traits deemed valuable while removing those deemed incompatible. Not a civilization that arose naturally through gradual cultural evolution. A project, an experiment in genetic engineering at civilizational scale, running for 600 years, achieving astronomical and mathematical sophistication that exceeded contemporary older world civilizations, building monuments that still stand, creating knowledge systems that we're only recently learning to fully appreciate, and then collapsing completely within decades once the social structures maintaining the genetic program fell apart. That's darker than we imagined, not because the Maya were evil or because genetic engineering is inherently wrong. It's darker because it forces us to confront what civilizational achievement actually costs. The Maya show us that remarkable intellectual and technological sophistication can coexist with systematic violence, that breeding programs producing enhanced populations require eliminating people who don't fit the genetic plan, that concentrated knowledge and concentrated genetics often go together because both require selection, organization, and willingness to make hard choices about what gets preserved and what gets removed. We want to believe that great civilizations arise through noble means, that achievement reflects enlightenment, that societies capable of sophisticated mathematics and astronomy must also be ethically advanced. The Maya evidence says otherwise. They were brilliant and brutal, sophisticated and savage. They understood heredity well enough to implement genetic selection across centuries while lacking the ethical frameworks that modern societies claim would prevent such programs. Or maybe they had ethical frameworks that justified systematic elimination as necessary for civilizational benefit. Maybe they weighed costs against achievements and decided the trade-off was acceptable. We're making similar calculations now, not through centralized government programs imposing eugenic selection, through distributed individual choices in fertility clinics, through national research priorities, funding genetic enhancement projects, through regulatory frameworks that permit or encourage trait-based embryo selection, through market forces creating demand for genetic advantages parents can purchase for children. Different mechanisms, same outcome. Shifting human population genetics away from natural diversity toward designed optimization. The question the Maya evidence forces us to answer is whether that shift is sustainable, whether genetically engineered populations prove stable over centuries, or whether they carry inherent fragilities that natural populations don't, whether the benefits of enhancement justify the costs in violence, stratification, and eventual collapse, whether modern technology really does provide safeguards that ancient methods lacked, or whether the fundamental problems with genetic engineering transcend technical sophistication. We won't know the answer until we've conducted the experiment and observed outcomes over multiple generations. The Maya took 600 years to run their experiment. We might get results faster because modern populations are larger. Genetic changes can be implemented more rapidly, and monitoring technologies allow us to track outcomes in real time rather than waiting for archaeological discovery 1,200 years later. But faster doesn't mean soon. Even optimistic timelines put measurable outcomes decades away. Comprehensive understanding of long-term consequences might take centuries, which means the decision to proceed with genetic enhancement is being made with fundamentally incomplete information. We know it's technically possible because the Maya demonstrated that. We know it can produce benefits because their astronomical and mathematical achievements are documented. We know it carries costs because their mass graves and elite commoner genetic stratification are documented. We know it eventually failed because their civilization collapsed. What we don't know is whether modern implementations can achieve benefits while avoiding costs and preventing failure. That uncertainty should make us cautious, but caution competes with other pressures. International competition between nations developing enhancement technologies, parental desire to give children advantages, scientific ambition to push boundaries of what's possible, commercial incentives to profit from reproductive technologies. All of these forces drive toward rapid implementation regardless of uncertainty about long-term consequences. The Maya might have faced similar pressures: competition between city-states, elite desire to produce superior offspring, priestly knowledge about heredity, creating temptation to apply that knowledge systematically. We don't know what motivated their genetic program because the texts explaining purposes and justifications haven't been translated yet. The December excavation might reveal inscriptions describing why they did it, or the inscriptions might focus on technical details about how they did it without explaining motivations. Or the sealed chambers might not contain textual explanations at all, just evidence of what was done. Even without knowing Maya motivations, we can observe outcomes. Enhanced populations achieved remarkable things. The astronomical observations, the mathematical notation, the architectural precision, these weren't accidents. They required cognitive capabilities that genetic selection might have concentrated. But those same populations proved fragile. When environmental stress hit, when social structures fractured, when the institutional machinery maintaining breeding programs collapsed, the carefully engineered civilization disintegrated within decades, faster than most civilizations fall, suggesting that genetic engineering might accelerate both achievement and collapse. That acceleration matters for planning modern programs. If genetically engineered civilizations follow boom-bust cycles where rapid achievement leads to rapid failure, then enhancement might provide short-term benefits at the cost of long-term instability. Good for individual nations competing over decades. Bad for human civilization enduring over centuries. The optimization problem isn't just how do we enhance capabilities, but how do we enhance capabilities while maintaining stability across timescales longer than individual lifetimes or national political cycles. The Maya apparently didn't solve that problem. Or maybe they did, and the collapse was intentional, a planned endpoint after knowledge preservation objectives were met. The texts in sealed chambers might clarify whether failure was unexpected catastrophe or designed completion. Either interpretation has implications for modern programs. If collapse was unexpected despite sophisticated understanding of genetics and heredity, that suggests inherent instabilities that knowledge alone can't prevent. If collapse was planned, that suggests Maya genetic engineering was never meant to create permanent civilization, but rather temporary repository of knowledge designed to last specific duration, then deliberately shut down. The second interpretation sounds like science fiction, but it's not impossible. If the environmental catastrophe around 1200 BCE was severe enough that knowledge preservation became primary concern, if multiple civilizations were organized as redundant archives designed to survive until conditions improved and future societies could reconstruct what was lost, then planned termination makes sense. Preserve knowledge for specific duration. Encode it in genetics, monuments, texts, astronomical observations. Wait for future civilization to develop technology to read the encoding. Then shut down because the preservation phase is complete, and whatever comes next should build on preserved knowledge, rather than continuing archaic organizational structures. We might be the future civilization they were preserving knowledge for. The timing is suspicious. Just as we develop capability to sequence ancient DNA, translate inscriptions, analyze astronomical observations, understand genetic engineering, the evidence from multiple ancient civilizations becomes available, showing they all implemented similar programs during the same centuries after the same catastrophe, as if the whole thing was coordinated preservation project with built-in time release, calculated so that evidence would surface when needed to inform contemporary decisions about genetic enhancement. Or it's just coincidence. Archaeological discoveries happen when they happen, technological advances occur on their own timelines. The synchronization between when we're debating genetic enhancement and when we're discovering that ancient civilizations attempted it might be random correlation rather than intentional design. Probably is random. The alternative requires assuming planning and coordination extending across millennia, organizational sophistication that seems implausible for societies we consider technologically inferior to ours. But the genetic data says those societies weren't technologically inferior in understanding heredity. They implemented selection programs that modern population geneticists recognize as sophisticated, even though they lacked molecular biology. They understood trait inheritance, family lineage tracking, long-term genetic planning, maintenance of breeding programs across generations. That's not primitive knowledge. That's applied genetics at civilizational scale. If they could do that, what else might they have been capable of that left no obvious archaeological trace? Questions we can't answer with current evidence. Questions that might never be answerable if critical information was lost during collapse or deliberately destroyed. Questions that the sealed chambers might address if they contain comprehensive documentation rather than fragmentary glimpses. 10 days until the first chamber opens. We'll know soon whether Maya builders left clear explanations or whether we're left interpreting ambiguous evidence that supports multiple competing theories. Either way, the genetic data is solid. Trans-Pacific populations integrated into Maya civilization, systematic breeding programs concentrating specific traits, mass graves documenting elimination based on ancestry, cognitive enhancement visible in genomic selection signatures, multi-continental population assembly following global environmental catastrophe. All of this is documented, replicated, peer-reviewed. Not speculation, not fringe archaeology. Mainstream ancient DNA analysis producing results that force us to completely revise understanding of Maya origins and development. That revision has implications beyond Maya studies. If one civilization was engineered through population assembly and genetic selection, were others? The preliminary data from Egypt, Mesopotamia, Indus Valley, and China suggests yes. The same patterns appearing globally during the same time windows, which means human civilization as we understand it might not be the product of isolated populations independently developing complexity. It might be deliberate construction project involving population movement, knowledge transfer, and genetic engineering on scales we're only beginning to appreciate. Who organized it? That's the question everything else depends on. If ancient civilizations independently figured out population genetics and all happened to implement similar programs during the same centuries, that's one scenario. If they were all following instructions from predecessor civilization that survived the catastrophe and used its knowledge to rebuild human societies with enhanced populations, that's a completely different scenario. The evidence we have doesn't distinguish between these possibilities. Both fit the genetic data. Both explain the synchronization across civilizations. Both account for knowledge systems appearing without obvious local developmental sequences. The sealed chambers might provide clarity. If texts describe origins of the genetic program, if they name the source of knowledge about heredity and population assembly, if they explain who taught the first fathers and where that teaching came from, then we'll know whether Maya civilization was independent development or part of larger coordinated effort. That knowledge changes everything about how we interpret human history and what we should expect about civilization collapse patterns. Because if civilizations are engineered projects with design durations, then collapse isn't failure, it's completion. Planned obsolescence, ensuring that knowledge gets preserved, but populations don't persist indefinitely in configurations optimized for conditions that no longer exist. And if multiple ancient civilizations all collapse during narrow windows after achieving specific levels of sophistication, that pattern might not be coincidence. It might be design feature. Build civilization, preserve knowledge, shut down before engineered populations become maladapted to changing conditions, leave evidence for whoever comes next. That interpretation makes Maya collapse less mysterious. It wasn't environmental catastrophe or social collapse or warfare causing unexpected failure. It was design termination once knowledge preservation objectives were met. The mass graves showing systematic elimination might not be evidence of program failure, but evidence of program conclusion. Cleaning up, removing engineered populations that were never meant to persist after their knowledge preservation function was complete. Brutal, but consistent with civilization organized around functional objectives rather than perpetual existence. We don't want that to be the answer. We want civilizations to be eternal human endeavors, rising and falling through normal historical processes, leaving ruins as testament to achievements that time eventually wore away. The alternative, that civilizations might be temporary projects with designed terminations, suggests we're not in control the way we think we are. That human societies might serve purposes beyond the understanding of populations living in them. That what we consider civilizational collapse might sometimes be planned conclusions rather than catastrophic failures. The Maya evidence doesn't prove this interpretation, but it's compatible with the data. And if it's true, if ancient civilizations were engineered knowledge preservation projects, then modern civilization might be the same thing, built on foundations we don't fully understand, incorporating genetic enhancements we're only recently discovering, following trajectories designed by whatever preceded us, approaching potential termination points that will look like collapse from inside, but might be completion from outside. Paranoid thinking. Probably easier to assume ancient civilizations arose and fell through natural processes than to imagine multi-millennial planning spanning catastrophes and coordinating genetic engineering across continents. Easier to interpret Maya collapse as failure than as designed completion. Easier to believe we're in control of our civilization's trajectory than to suspect we're following patterns established long before we existed. But the genetic data doesn't support easy interpretations. It supports uncomfortable ones. Trans-Pacific populations appearing in Mesoamerica after global catastrophe. Simultaneous implementation of similar genetic programs across multiple civilizations. Knowledge systems emerging without developmental sequences. Collapse patterns showing suspicious synchronization. All of this requires explanations that current paradigms don't accommodate, which is why the next 5 years matter so much. The sealed chambers opening, the comprehensive genetic datasets being published, the analytical tools improving, the evidence accumulating. By 2030, we'll have the most complete picture of ancient population genetics ever assembled, enough data to detect patterns invisible in current limited samples, enough temporal resolution to track genetic changes with generational precision, enough geographic coverage to compare programs across civilizations and see whether they were independent or coordinated. And that comprehensive picture will inform decisions about whether modern civilization should implement genetic enhancement. Not in abstract ethical debates, in concrete policy choices about funding, regulation, implementation. Do we proceed knowing that ancient civilizations attempted this and collapsed? Do we pause until we understand what caused previous failures? Do we rush ahead in competitive races where hesitation means falling behind rivals willing to take risks where not? Those decisions are being made now, not waiting for comprehensive Maya evidence, not waiting for long-term outcomes from programs already underway. Made in real time by people with incomplete information and competing pressures. The Maya at least had centuries to observe outcomes before their program collapsed. We're implementing ours while still excavating evidence from previous attempts, hoping that our superior technology provides advantages they lacked, assuming that understanding molecular mechanisms prevents instabilities they couldn't avoid, believing that modern ethics creates safeguards they didn't have. Maybe we're right. Maybe modern genetic enhancement succeeds where ancient programs failed. Maybe comprehensive understanding of genomics provides advantages that observational genetics couldn't. Maybe distributed individual choices produce better outcomes than centralized institutional programs. Maybe the Maya collapsed for reasons unrelated to genetic engineering, and we're drawing false lessons from their trajectory. Or maybe we're repeating their mistakes with modern tools. Maybe the problems with genetic engineering are inherent rather than technical. Maybe reducing population genetic diversity creates fragility, regardless of how sophisticated the reduction process is. Maybe social stratification between enhanced and unenhanced populations generates instability that technology can't prevent. Maybe civilizations built on genetic engineering are inherently temporary, regardless of the mechanisms used to engineer them. We'll find out. The experiments are running. The genetically enhanced populations are being born. The data about outcomes will accumulate over decades, and by the time we know for certain whether modern genetic enhancement succeeds or fails, whether we've avoided Maya failure modes or repeated them, millions of genetically modified humans will exist. Permanent alterations to the human gene pool based on decisions made with incomplete information and inadequate understanding of consequences. That permanence is the scariest part. The Maya program was reversible. Once the institutional structures maintaining breeding selection collapsed, natural reproduction reasserted baseline population genetics within a few generations. Modern germline edits are forever. CRISPR modifications pass to all descendants automatically. No reversion mechanism. No going back if we discover we've made terrible mistakes. The genetic changes we're implementing now will persist for thousands of years, spreading through human populations, affecting descendants who never consented to being engineered. The Maya gave us data. They documented what they tried, how long it worked, what it cost, and how it ended. They preserved that documentation in formats that would survive collapse and eventual discovery. They left warnings or instructions, or just evidence for whoever came next. to analyze and learn from. We're analyzing, we're learning. But are we learning the right lessons? Are we learning that genetic enhancement is possible and we should attempt it with better tools? Or are we learning that genetic enhancement is dangerous and we should avoid it despite the tools? Are we learning that ancient failures predict modern failures? Or are we learning that ancient failures teach us how to succeed? The same evidence supporting opposite conclusions depending on what questions we prioritize and what values we bring to interpretation. The December chamber opening won't settle these debates. It will provide more evidence, more data about what the Maya did and potentially why they did it. But interpretation remains contested. Enhancement advocates will emphasize achievements. Critics will emphasize costs. Policymakers will make decisions based on national interests rather than long-term civilizational stability. Parents will make choices based on immediate advantages for children rather than population-level consequences across centuries. And somewhere in Guatemala, beneath a temple built by populations assembled from across the planet and genetically engineered over centuries, stone tablets wait to deliver whatever message the Maya chose to leave. Instructions, warnings, explanations, documentation. We're about to read that message. Whether we're capable of understanding what it says about the true nature of human civilization, about the costs of achievement, about the relationship between genetic engineering and civilizational collapse, about whether we should proceed with confidence or caution. That's the real question. The Maya told us their story. They preserved it across 12 centuries. They encoded it in genetics and monuments and texts and astronomical observations. They left evidence distributed across disciplines, so that full reconstruction requires synthesizing archaeology, genetics, linguistics, astronomy, anthropology. They made it hard. Comprehensive understanding requires decades of excavation, analysis, publication, debate, synthesis. We're in the middle of that process, just beginning to see the complete picture, just starting to understand what they were and what they did and what happened when their engineered civilization encountered conditions it couldn't adapt to. And the universe doesn't care whether we learn from their experience or ignore it. The universe operates according to physical laws indifferent to human choices. But we care, or we should, because the decisions we're making now about genetic enhancement determine what human populations look like for thousands of years, whether we engineer better humans or create fragile populations vulnerable to collapse, whether we achieve temporary sophistication at the cost of long-term instability or maintain natural diversity and accept evolved capabilities, whether we repeat the cycle the Maya documented or write a different story with different ending. The choice is ours. The Maya gave us their data. The tools exist to analyze it. The evidence is comprehensive. The implications are clear. What we do with that information determines whether human history extends patterns established 3,000 years ago or breaks them, whether civilization in the 21st century learns from the past or becomes another sealed chamber waiting for excavation by whatever comes after us. December the 25th, 2025. The first chamber opens. March 2026. Comprehensive genetic results publish. By 2030, we'll know more about Maya population genetics than any ancient civilization ever studied. And then we decide, informed or not by their precedent, aware or not of the risks they documented. willing or not to learn from civilizations that attempted what we're attempting and left ruins as testament to what happens when genetic engineering meets civilizational ambition, meets systematic violence, meets eventual collapse. The DNA revealed the true origins of the Maya. They came from everywhere. They were assembled deliberately. They were genetically engineered systematically. They achieved remarkable sophistication. They paid horrific costs. And they collapsed completely. That's darker than we imagined. Not because the Maya were monsters, but because they show us what we might become if we followed the same path with modern tools, what humanity might sacrifice in pursuit of enhanced capabilities, what civilizations built on genetic engineering eventually face when the engineering proves less stable than the architects intended. The message is waiting. The chambers are opening. The choice is ours. Right now, as you listen to this, laboratory technicians at 3 separate ancient DNA facilities are processing samples that could rewrite everything we thought we knew about human civilization. Excavation teams in Guatemala are preparing equipment for the December 25th chamber opening. Epigraphers are reviewing Maya glyph databases, preparing to translate whatever inscriptions emerge. Geneticists are finalizing protocols for rapid DNA extraction and sequencing. Bioethicists are drafting position papers about how ancient genetic engineering evidence should inform contemporary enhancement policy. All of this happening simultaneously, while most of the world remains unaware that human civilizations have attempted genetic modification before, with documented outcomes waiting in sealed chambers and ancient bones. The disconnect between what researchers know and what the public understands creates information asymmetry that shapes policy debates in dangerous ways. Enhancement advocates can argue that genetic modification is untested speculation because most people don't know about Maya population genetics. Critics warn about unforeseen consequences without being able to point to historical precedent because that precedent isn't widely known. Policymakers draft regulations without comprehensive understanding of what happened when previous civilizations attempted similar programs. The evidence exists. The analysis has been conducted. The publications are available. But they're trapped in specialist literature that broader audiences don't access, which means decisions about humanity's genetic future are being made by populations who don't know their ancestors already conducted these experiments, who don't understand that genetic enhancement isn't revolutionary progress but revival of ancient practices, who can't evaluate whether modern technology actually provides advantages that ancient methods lacked because they don't know what ancient methods achieved or how they failed. The information exists. It's just not reaching the people whose lives will be affected by decisions made based on incomplete information. That's why comprehensive Maya evidence needs to emerge soon, not just for academic interest, for immediate policy relevance. The programs implementing genetic enhancement are accelerating. Chinese research expanding. Fertility clinics normalizing trait-based selection. Regulatory frameworks being drafted without full consideration of historical outcomes. If Maya data remains obscure until after these programs are established and normalized, historical precedent arrives too late to influence decisions. The experiment is already running. Genetically modified populations already being born. By the time comprehensive analysis is published and widely discussed, millions of enhanced individuals will exist reversing course becomes politically, ethically, economically impossible. The timing matters. December 2025. Chamber opening. March 2026. Genetic results. These dates aren't arbitrary. They represent when evidence becomes comprehensive enough, public enough, dramatic enough to force broader discussion. When discoveries can't be dismissed as specialized research, irrelevant to contemporary policy. When the connection between ancient genetic engineering and modern enhancement programs becomes undeniable even to audiences who don't follow population genetics literature, assuming the evidence is what researchers expect, assuming the sealed chambers contain comprehensive documentation rather than fragmentary glimpses, assuming the inscriptions are decipherable and explicit rather than ambiguous and metaphorical, assuming the genetic data shows clear selection signatures rather than patterns that multiple competing theories can explain. None of this is guaranteed. Archaeological discoveries often raise more questions than they answer. The chamber might contain preservation failures, degraded materials, ambiguous contents that don't provide the clarity everyone's hoping for, or it might contain exactly what researchers expect. Stone tablets with detailed inscriptions describing the genetic program's implementation, purposes, and outcomes remains showing the pinnacle of 600 years of systematic selection. Artifacts embodying technologies or knowledge systems deliberately preserved separately from materials in active use. Astronomical observations encoded in chamber alignments revealing sophistication that current understanding underestimates. Everything necessary to fully reconstruct what the Maya were doing, why they were doing it, and what happened when their engineered civilization encountered conditions it couldn't adapt to. That best-case scenario provides unprecedented historical precedent for contemporary genetic engineering debates. Complete documentation of a civilization that attempted population enhancement, maintained it across centuries, achieved remarkable results, paid systematic costs, and eventually collapsed when the program proved unsustainable. Not theoretical modeling of what might happen, actual historical outcomes from a society that ran the experiment and documented everything. That documentation will either validate enhancement advocates who argue that genetic selection can produce superior populations, or vindicate critics who warn that population engineering creates instabilities leading to collapse, or more likely, it will provide evidence supporting both positions. Yes, enhancement works in the sense of producing measurable improvements in targeted capabilities. Yes, enhancement carries costs in systematic violence and social stratification. Yes, enhanced populations achieve things unenhanced populations might not. Yes, engineered civilizations prove fragile in ways natural populations aren't. All of these can be simultaneously true. All of these might be inseparable consequences of the same process, which means the Maya evidence won't settle debates. It will sharpen them, give both sides concrete historical examples rather than theoretical speculation, provide data about what happens when genetic engineering runs for 600 years, rather than models predicting what might happen if we implement it now. Ground discussions in documented outcomes from previous attempts rather than assumptions about untested possibilities. And that grounding matters enormously for policy formation. Current debates operate largely in absence of historical precedent. We argue about whether genetic enhancement is ethical, without examples of societies that attempted it and recorded consequences. We debate whether benefits justify costs without data about actual benefits and actual costs from previous programs. We predict what might happen based on genomics theory and evolutionary biology without studying what did happen when ancient civilizations applied genetic knowledge at population scales. The Maya provide that historical data, or they will once the evidence is fully excavated, analyzed, and published. The December chamber opening accelerates the timeline. Instead of waiting years for conventional archaeological publication, the findings will be analyzed immediately, streamed to partner institutions, processed in real time, and made available within months rather than years. That compression brings historical precedent into contemporary policy discussions, while those discussions are still shaping initial frameworks rather than after decisions have already been made, and implementation is too advanced to modify. Unless the evidence is ambiguous, unless the inscriptions don't clearly explain purposes and methods, unless the genetic data shows patterns that multiple interpretations can accommodate, unless what researchers find raises more questions than it answers about Maya, motivations, knowledge sources, organizational structures, and collapse mechanisms. Archaeological reality often disappoints expectations. The sealed chamber might contain less information than hoped, might preserve poorly, might require years of careful analysis before yielding interpretable results. But even ambiguous evidence moves discussions forward. Even fragmentary data provides more than we currently have. Even unclear inscriptions might contain details that illuminate aspects of Maya genetic engineering that current publications don't address. The chamber opening matters not because it will definitely provide comprehensive answers, but because it represents concentrated effort to extract maximum information from a context deliberately designed for knowledge preservation. Maya builders sealed that chamber for a reason. They chose specific contents. They positioned it at the architectural center of their most important city. They preserved it through collapse and centuries of abandonment. Whatever is inside represents what they considered most important to preserve for future discovery. That curatorial decision, that choice about what to include in a time capsule designed for audiences centuries or millennia in the future, reveals priorities. What knowledge mattered enough to protect? What information did they think future civilizations would need? What warnings or instructions or explanations did they consider crucial to communicate? The contents answer these questions, even if the specific details are ambiguous. The act of preservation itself tells us something about how Maya builders understood their civilization's purpose and what messages they wanted to transmit. Modern civilizations are making similar curatorial decisions now, repositories being designed to preserve information across millennia, Arctic Seed Vault protecting genetic diversity of food crops, multiple projects documenting human knowledge in formats expected to survive civilizational collapse. The Maya did this 1,200 years ago. We're doing it now, and the fact that we're both engaging in long-duration knowledge preservation suggests that civilizations reaching certain levels of sophistication recognize their own fragility, understand that what they build might not last, want to ensure that if they collapse, something survives for whoever comes next. That recognition of fragility is important. It means the Maya knew their civilization was vulnerable, knew that genetic engineering programs creating enhanced populations also created dependencies and instabilities that might eventually cause failure. And they chose to preserve evidence about what they'd done, how they'd done it, and what happened, suggesting they wanted future civilizations attempting similar programs to learn from their experience rather than repeating their mistakes. Or they just wanted to preserve their achievements, show future societies what they'd accomplished, document their sophistication for audiences that would appreciate it. The motivations might be simple pride rather than pedagogical concern, wanting credit, wanting recognition, wanting acknowledgement that they'd existed and achieved remarkable things, even though their civilization collapsed and their cities were abandoned. Human motivations rarely reduce to single simple drivers. The Maya probably sealed chambers for multiple overlapping reasons, some practical, some symbolic, some we wouldn't understand without cultural context we've lost. But regardless of motivations, the preservation happened. The evidence exists. The chambers are being opened. The genetic data is accumulating. The analysis is proceeding. And whether the Maya intended to teach us lessons or just wanted to document their accomplishments, we're receiving information about genetic engineering at civilizational scale from a society that attempted it and left comprehensive documentation. What we do with that information is the choice that matters. Do we proceed with modern enhancement programs informed by Maya outcomes? Do we design implementations that avoid their failure modes? Do we maintain genetic diversity while attempting enhancement? Do we build social structures that can handle stratification? Do we monitor carefully for early warning signs of the kind of instability that preceded their collapse? Or do we dismiss their experience as irrelevant to modern capabilities? Assume our technology makes us immune to problems they couldn't solve? Proceed without caution because we're confident we're smarter than our ancestors? History suggests the second path is more likely. Civilizations rarely learn from predecessors' mistakes. More often, they assume they're special, that circumstances are different, that superior knowledge prevents previous failures. The Maya probably thought the same thing. Whatever predecessor civilization taught them genetic engineering likely also collapsed. The Maya might have believed they could succeed where previous attempts failed. And for 600 years, they did succeed. Until they didn't. Until the instabilities their genetic program created overwhelmed adaptive capacity and everything fell apart. We're walking the same path, believing we're different, assuming modern genomics prevents ancient mistakes, confident that understanding molecular mechanisms provides advantages that observational genetics couldn't. Maybe we're right. Maybe comprehensive knowledge of heredity at molecular level really does allow enhancement without the instabilities that destroyed previous attempts. Or maybe the problems with genetic engineering are inherent in the process rather than the implementation details. Maybe reducing population genetic diversity creates fragility, regardless of how sophisticated the reduction methods are. Maybe we're just sophisticated enough to document our failure more comprehensively than the Maya documented theirs, leaving better evidence for whatever civilization comes after us to excavate and debate whether to repeat the cycle again. The universe doesn't care which scenario plays out. Physical laws operate indifferent to human ambitions. Genetic drift, selection pressures, population dynamics. These follow mathematical principles that apply equally to ancient and modern populations. The Maya were subject to those same principles we are. Their genetics work the same way ours does. The outcomes they experienced result from processes that will produce similar outcomes if we implement similar programs. Unless modern technology really does provide game-changing advantages, unless understanding molecular mechanisms allows unprecedented precision, preventing instabilities, unless we're genuinely different in ways that matter for genetic engineering success, we'll find out the experiments are running, the populations are being modified, the outcomes will become measurable over decades, and by the time we know for certain whether modern enhancement succeeds or fails, the genetic changes will be irreversible, spread through human populations, affecting descendants for thousands of years, based on decisions made with incomplete information while evidence from previous attempts was still being excavated and analyzed. That's the scary part. Not that we're attempting genetic enhancement, that we're attempting it while simultaneously discovering what happened the last time civilizations tried it. The Maya evidence is arriving too late to inform initial implementation decisions, but early enough to reveal problems with programs already underway. The timing is almost perfectly wrong for using historical precedent to prevent repeating historical mistakes. Unless the December chamber changes that timeline, unless the findings are dramatic enough, clear enough, publicized enough that they force immediate policy discussion, unless stone tablets with explicit warnings about genetic engineering dangers prove persuasive enough to pause programs mid-implementation, unless genetic data showing Maya cognitive enhancement followed by civilizational collapse convinces enhancement advocates to proceed more carefully, unless something in that chamber makes the connection between ancient genetic engineering and modern programs so undeniable that even politically and commercially motivated actors recognize the need for caution. That's a lot of unlesses, relying on archaeological discovery to reshape policy trajectories already in motion, driven by powerful interests and institutional momentum, hoping that evidence about events 100 years ago proves compelling enough to override contemporary pressures, assuming that people making decisions about genetic enhancement care about what happened to the Maya and see their civilization's trajectory as relevant to modern programs rather than as interesting historical curiosity without contemporary implications. Maybe it happens. Maybe the evidence is compelling. Maybe the connections are undeniable. Maybe policymakers incorporate Maya outcomes into enhancement regulations. Maybe parents choosing whether to genetically modify embryos consider what happened when ancient civilizations implemented population-scale selection. Maybe enhancement advocates design programs that consciously avoid failure modes documented in Maya genetic data and mass graves and civilizational collapse. Or maybe none of that happens. Maybe the chamber opens, the evidence gets analyzed, the publications appear, and policy continues unchanged because historical precedent proves less influential than competitive pressures, commercial incentives, and confidence that modern technology makes ancient warnings irrelevant. Maybe we proceed with genetic enhancement despite Maya evidence because we believe we're different, we're smarter, we won't make their mistakes. Maybe we repeat their trajectory and leave our own ruins for excavation by whatever civilization emerges after ours collapses, armed with even better technology and the same confidence that they won't repeat our mistakes because they're special. The cycle might be fundamental. Civilizations reaching capability to modify genetics attempt it, achieve temporary enhancement, create instabilities, collapse, leave evidence for next civilization. Repeat. The Maya might just be our turn to discover evidence from the previous cycle. Our descendants might be the next civilization discovering our evidence and debating whether to repeat the cycle again. Maybe that's how it works. Maybe genetic engineering and civilizational collapse are inevitably linked regardless of technological sophistication. Maybe we're not learning from the Maya. We're just gathering data for whoever comes after us. Dark thought, depressing possibility, but consistent with human history, showing civilizations rarely learning from predecessors, consistently repeating mistakes, believing they're exceptional, assuming their circumstances are different, proceeding with confidence into disasters that retrospectively seem predictable. The Maya probably knew about civilizations that collapsed before them, probably thought they could avoid those failures, and they did for 600 years, until they didn't. We know about the Maya now. We're analyzing their evidence. We're understanding what they did and what happened. And we're proceeding with genetic enhancement programs anyway, because we think we're different, because we have better technology, because we believe molecular biology provides advantages that observational genetics lacked. Maybe we're right. Maybe this time really is different. Or maybe we're just the current iteration of a pattern that's been repeating for thousands of years across civilizations that all thought they were special. The December chamber might clarify, might contain evidence showing whether the Maya were part of a longer cycle, whether they knew about predecessor civilizations that attempted genetic engineering before them, whether they saw themselves as continuing a pattern or breaking it, whether they left instructions about how to succeed where they failed, or warnings about why failure is inevitable, whether their collapse was unexpected catastrophe or designed completion of temporary knowledge preservation phase. Or the chamber might just add more data without answering fundamental questions about whether genetic engineering at civilizational scale can succeed long-term, or whether it inevitably leads to collapse. Either way, the evidence will be more comprehensive than what we currently have. The analysis will be more detailed, the publications will be more thorough, and the question of what we do with that information will become more urgent as genetic enhancement programs expand and genetically modified populations grow. 10 days until the chamber opens. 3 months until comprehensive genetic results publish. 1 year until integrated analysis appears, synthesizing genetic, archaeological, linguistic, and astronomical evidence into coherent models of what Maya civilization was and what happened when it collapsed. 2 years until policy frameworks incorporating historical precedent are drafted and debated. 5 years until we know whether those frameworks actually influenced enhancement programs or whether implementation proceeded regardless of historical warnings. That's the timeline. Evidence emerging, analysis proceeding, publications appearing, policy discussions incorporating or ignoring historical data, programs continuing or pausing based on whether Maya outcomes prove persuasive. genetically modified populations growing regardless, because even if programs pause, individuals already born carry modifications that will pass to descendants. The genetic changes are happening now, irreversibly, permanently altering human populations based on decisions made before comprehensive evidence about previous attempts was fully analyzed. And somewhere in Guatemala, beneath a temple built by populations assembled from across the planet and genetically engineered across centuries, stone tablets covered with inscriptions wait to deliver whatever message the Maya chose to leave. A message preserved through collapse and abandonment. A message encoded for discovery by whatever civilization developed the technological capability to read genetic sequences and translate ancient texts and understand what genetic engineering looks like in archaeological data. That civilization is us. We're reading the sequences, we're translating the text, we're understanding what genetic engineering produces in population genetics and skeletal remains and social stratification. We're analyzing the evidence and debating what it means and deciding whether to proceed with our own enhancement programs informed or not by their precedent. The Maya left us their story, not in mythology, in DNA, in bones, in mass graves showing systematic elimination, in elite burials showing concentrated foreign ancestry, in texts describing distant homelands and catastrophic floods, in astronomical alignments encoding sophisticated knowledge, in sealed chambers waiting 200 years for opening. They documented what they did, how long it worked, what it cost, and how it ended. They preserved that documentation through civilizational collapse. They left it for us. Whether we use it wisely or ignore it in our confidence that we're different, that's the question the next decade answers. Whether we learn from civilizations that attempted genetic engineering before us or repeat their mistakes with modern tools, whether we avoid their failure modes or discover that failure is inherent regardless of technological sophistication, whether we write a different ending or extend the same story they told 1,200 years ago. The choice is ours. The Maya gave us data. The universe doesn't care what we do with it, but we should care, because the decisions we're making now determine what human populations look like for thousands of years, whether we maintain natural genetic diversity or optimize for traits we think beneficial, whether we build stable civilizations or fragile ones, whether we achieve temporary sophistication at the cost of long-term collapse or find ways to enhance capabilities while maintaining adaptability. The evidence is waiting. The chambers are opening. The analysis is proceeding. And the question isn't whether we'll finish reading what the Maya left. The question is whether we'll accept what it says about genetic engineering, civilizational ambition, systematic violence, remarkable achievement, and inevitable collapse. Whether we'll see their trajectory as warning or blueprint. Whether we'll choose differently or repeat the cycle. December 25th, 2025. The chamber opens. March 2026. The genetics confirm. And somewhere between excavation and analysis and publication and policy and implementation, we decide what kind of civilization we're building. One that learns from the past, or one that becomes the past's next iteration, one that breaks the cycle, or one that extends it. The Maya are waiting to tell us which. We're about to find out if we're ready to listen.